All
Recording
right.
in progress.
Good morning. Good morning, good afternoon, and good evening, wherever you are in this world. Welcome, everybody, to the 2023 Analysis Working Group Symposium. Before we begin the event, I just wanted to cover a few housekeeping items. First and foremost, I wanted to give a shout out and a big thank you to Blue Marble Space, who is sponsoring the event and providing the platform that we are using. I also wanted to give a big thank you to Marco Bolt for his stellar support for this event. As we proceed through the event, and if you're really interested in the work that is happening within the analysis working groups, we highly encourage you to join. Marco will put up a link in the chat for a sign-up form, which is going to look like this. And if you fill it out, I will send that information to each of the respective AWG leads. If in case you happen to miss signing up for the form, don't worry. We'll put up this form throughout the event. And you can also find the form on our website at osdr.nasa.gov/bio/AWG/join. So for all of the participants, your audio and video is disabled. And if you have any questions for the speakers, then we'll be using Conference.io, and Marco will put the link to that to Conference.io in the chat. For those of you that are new to Conference.io, the way that you can ask a question is by first clicking on this plus sign here, which will allow you to put in a question. Then you're going to type in your question. And lastly, you can use the caret to upvote any questions you may like. I will do my best to try and answer as many questions as possible in the allotted time. But to help me prioritize which questions get asked, I will be asking the questions that have the highest number of upvotes. So with that, I would like to hand it over to Lauren Sanders, who is the acting project scientist for GeneLab.
Thank you, Lavorka. Welcome, everyone. So nice to see you here. Can you go to the next slide, Lavorka? So the work that we're going to hear about at today's symposium focuses on space biology, which aims to pursue safe and successful space exploration by studying how these basic life processes are affected by the space environment. Next slide. NASA has several open science resources to maximize this spaceflight research. So for example, the Biospecimens Sharing Program dissects and preserves rodent tissues from flight and ground investigations and coordinates tissue sharing. NBISC maintains a collection of non-human tissue and microbial specimens, which can then be requested by researchers. ALSDA collects and curates mission and project imaging data, while the GeneLab Data Repository collects and curates spaceflight and space-relevant omics data. Next slide. Recently, the two data repositories, ALSDA and GeneLab, along with the Biospecimen Repository, NBISC, have merged to form the NASA Open Science Data Repository. This is now going to allow researchers to search across missions to find different phenotypic, omics data, and ultimately even the remaining biospecimens from specific animals. We implement a single submission portal to streamline the process of submitting data, which is also facilitated by a user interface for research data submission agreements, or RDSAs. And across the entire database, we're working to ensure that data are maximally fair, findable, accessible, interoperable, and reusable. We have a new OSDR landing page where you can browse the data repository or access the submission portal, read more about these open science projects, or see our recent news and release data sets. The OSDR ecosystem, you can see on the right-hand side of this slide, is multifaceted. We have the data which are wrapped in an API for easy access. We also have the biological data management environment, as well as a workspace for data submitters, which uses standardized assay configurations for data submission, as well as common ontologies to strengthen metadata across all the assays. The GeneLab data processing team processes raw omics data and publishes standardized process outputs, which are also accessible on our repository user interface and the visualization and analysis platform. We also connect to various extramural data tools and databases. Next. Here's a view on the left-hand side of the user interface that I just mentioned for the research data submission agreement, where there are many new assays that have been implemented that you can select from to enter your data and all the parameters related to your experiment. We're also about to release an update to our visualization portal. You can see some screenshots on the right-hand side, and this update will enable visualizing gene expression data sets across emissions. We're also working on an environmental data application, which will allow users to compare environmental factors like CO2 and radiation levels across emissions. And we're just now starting the process of working on a machine learning modeling portal using OSDR data. We're currently in the process of ingesting the data from the Inspiration 4 mission with the appropriate access restrictions for sensitive data types. Notably, thanks to the hard work our curation team has put into developing reusable assay configurations, OSDR was already set up to receive and curate each data type. Overall, so far, 52 publications have been enabled by the OSDR system. And if you look at the graph on the top right, after the introduction of the analysis working groups, which we're going to hear from today in 2018, our user base grew significantly. And on the bottom right, after the introduction of process data and visualization, data downloads have nearly doubled. So in 2018, GeneLab formed a space biology community called the Analysis Working Groups of multiple hundreds of international scientists. The aim of the Analysis Working Groups, or AWGs, is to provide feedback on processing pipelines, essential metadata, while maximizing the scientific return on space experiments. We'll see talks from each AWG today. And as Lavorka mentioned, we'll put the link to the sign-up form in the chat if you'd like to join an AWG. So the first 4 AWGs that were initially formed had a focus on the molecular omics data types and are organized into separate focus areas: animal research, plant, microbes, and multi-omics. In 2021, a new community of researchers formed the ALSDA AWG, which focuses on standardizing physiological, phenotypic, and behavioral data. This uses the phenotypic data from the ALSDA. And then very recently, the Artificial Intelligence Machine Learning AI/ML AWG was created and focuses on reusing OSDR data for AI/ML applications. We published the AWG charter on our website, and I just wanted to highlight here that the purpose of these AWGs is to increase the effectiveness of the GeneLab and the OSDR data system and to maximize knowledge gain from all of these precious data sets. I also want to note that active participation in an AWG can lead to involvement in a scientific project and possibly authorship on a scientific paper. So at the symposium today, we have a packed morning agenda with a keynote address from Eliah Overbey, who will talk about the Inspiration 4 data and scientific presentations from AWG members. The afternoon agenda will provide us with continuing scientific presentations from AWG members, as well as status updates from each AWG. I will now pass on to Sylvain Costas, who's the project manager for Open Science for Life in Space.
Well, thank you, Lauren, for the introduction of Open Science. And then I'm going to talk a bit about Transform to Open Science to you guys. So there's really a big change happening right now for the Biological and Physical Science Division, which really has funded GeneLab for many years. So yeah, you can go to the next slide. It's fine. Thanks. And then SMD has this open source science initiative called OSSI. And if you click through the different activities we have, SPD41A is the data policy that has been put in place. 41A is the revised version from the 2019. We have Rosie's element. If you can click next, yeah, thank you. We have different Rosie's calls to support open science, and I'll talk a bit more about those grants coming up. transform to open science, that's something I'm going to talk a bit more about. But this year is the year of open science for not only NASA, but also the federal government at large. So, and then something that we are working actively on is the core services for science discovery. So the OSSI has a council and a working group at headquarters that some of us are part of. And we're building together this new ecosystem that really is supposed to be similar across all divisions. So BPS is one of the many divisions at SMD. You have planetary science, astrophysics, heliophysics, and so on. So we're just one small entity in this big ecosystem. Next slide. So talking about SPD 41A, that's the Scientific Information Policy. We revised it, the OSSI Council revised it and released a new version last year. There are some highlights that you need to be aware of because things have changed. So everything starts from that memo. So if you look on the left here, there's SPD 41A response to the new memo from OSTP. So the White House wanted to ensure free, immediate, and equitable access to federally funded research. The good news is that NASA was actively working on this before, so it didn't come as a surprise to us. And actually, it turns out SMD was very well ready for it, and SPD 41A was in the building when that memo came out. Some highlights so that you are aware of these changes as investigators. If you get funded by federal funding, especially for NASA, peer review publication needs to be openly available with no embargo period. The research data and software shared needs to be shared at the time of publication or at the very least at the end of the funding award. And in terms of more operational things, mission data need to be released as soon as possible, and unrestricted mission software developed are supposed to be open. And finally, we need to work on science workshop and meetings such as today to really broaden the participation of open science. Those are the big policies for the division. If you go for the directorate for SMD, but if you go to the next slide, the ALSDA Engine Lab, where you can go through all of them, so I can just walk through them. Thanks, that's good. We've been working actively on this policy and refining it for BPS, for our division. And so it looks very similar. You can see here the timeline. We and some of you were in the town hall that we have last year, but we did a public release of BPS back in November of 2022. Now we are at different phases. If you look at the timeline at the bottom, we are putting together FAQ. We are putting together a data management plan, and we call it the OSDMP because that's really the term that SMD is using. And that OSDMP, Open Science Data Management Plan, DS stands for Software and Data Management Plan. Those are really new items that we need to work on, and we're currently working with BPS to finalize some of these templates for the ROSIS call. And we're in the phase currently in implementing the policy for BPS. Once this is put in, the goal is to reach by 2025 to have full compliance from all the PI and all the different entities. So now, if you look at the highlights, it's very similar to SPD 41A, which is, at the top here, research data and software needs to be publicly available no later than the publication of the investigation results. Operation data should be immediately available to the public. And then any data that I'm not used to support publication must be submitted by the end of the period of performance, at the end of the grant. The way to retain some of your data behind a firewall is to ask for a no-cost extension, and there should be a good reason for it. So those are the main guidelines for the PI. On the data repo like GenLab and LSDA, we under very high rules. Basically any, some of you guys have worked on the augmentation data that we have generated at GenLab, and people enjoy this data. And as you know, for many years, we have already been following this kind of guidelines. The data are immediately available to you. There's never any proprietary time given to the data repo or the GenLab team. And so this is really trying to, we want full transparency and equity on access to data, and that's kind of like the goal. So this is the new ecosystem. And I know it's not news to most of you. You've heard me multiple times, but I wanted to insist. One little thing that's important, the GLab and ALSD has merged, and now you have a submission portal, a common submission portal. Any new PI who got NASA funding will use that system to actually do the research data submission agreement. So that used to be done by email, not anymore. It's all online and digital, and so we're very excited about this. So next slide. Okay, and then as you know, there's been quite a few calls on roses this year for open science, so I wanted to highlight some of them here. And there's a link. Go to Inspire and be familiar with F15, F2, and F8. For time's sake, we're not going to go into details. It's just there for your information. Next. So yeah, 2023 is a year of open science. What does it mean for TOPS? So TOPS is an initiative, driven by SMD at headquarters. Most centers are involved with this, and so there's a $40 million budget spread over 5 years and to really enable open science. So the goal is to release an open science curriculum, and we're working actively on this. Well, TOPS is not us, GLab or LSDA, but we provide some feedback. We want to engage the underrepresented communities in the advancements of open science. So it's one of the big goals. By creating a curriculum and teaching it, we hope to increase the concept of open science. And by the way, by 2027, we want to have, 2025, I forgot the time, but we want to have 20,000 people trained through this curriculum. So it's a very ambitious program. And so there are some, if you want to click on these different links at the bottom, please do so. Next. Why don't you go through everything? Yeah, thanks. So I would say one of the things that we have done recently that really highlight the concept of open science is really our education program with GLab for University. And we really took two different approaches. What we call the direct approach is teaching students directly, just like you've seen in the past with older programs like GLab for high school. We teach students, except they are typically undergrad or graduate students. And it started in 2021 with San Jose State University as a first pilot. We call those things student boot camps. This year in 2023, there will be a space bio and amplicon sequencing boot camp. In 2021, it was focused on RNA-seq. But the other thing that's a lot more exciting to me, I think, is really the GLab for You indirect approach, where we are training the educators. So it's an educator boot camp. And the beauty with this is once we train these educators on our tools, and they can go and teach their respective universities. So we had the first pilot last year in 2022. This was supported by JPL. It was very successful. There were 6 professors and 4 graduate students that took the class. They were, 4 of them were from HBCU and MSI. And then what was really exciting is last fall, one of these professors taught her class with the material we gave her. And then we are, we will repeat this experience very soon. So this is extremely exciting. The working group already heard about it on the left side, so I'm not going to repeat what Lauren just told you about it. So next. This is it for me. We are a bit ahead of schedule, which is great. And this is my great pleasure to introduce our keynote speaker. So if I can have the next slide. So Dr. Eliah Overbey is our keynote speaker today. She's a NASA postdoctoral fellow in the lab of Dr. Chris Mason at Cornell Medicine University. She got her PhD in genome science at the University of Washington, and she currently manages the Spaceomics and Medical Atlas, SOMA, to characterize multi-omics changes that occur in astronauts during spaceflight. I am very happy to have Eliah talk today, because I don't know if for us at GLab, we've known Eliah since 2018. She was one of the first, I think she was the first graduate student reaching out to me. We went to AWGs and we got the idea of the analysis working group in January, and I think in February she was already on it, calling me or reaching out to me saying, I want to be part of that. And she became a very essential person in the AWG. And in 2019, actually, she joined us for an internship with at GLab with Dr. John Galaska. So I'm so happy to see the progress of your career, Eliah, and I can't wait to hear you speak. So thank you.
Thank you so much, Slavan. Let me pull my slides up here. All right, thank you for inviting me. I'm excited to be speaking at the symposium today. Like Slavan had mentioned, I got my start in spaceflight research when I was in graduate school at the University of Washington. I joined the AWGs, the animal AWG. It's been really amazing to watch these groups grow over the years. It's grown so much in such a short period of time. So my talk today is about astronaut genomics and the age of commercial spaceflight. It's a bit future oriented, and it's looking at how we can use commercial spaceflight as an opportunity to perform more human multi-omic research. So what I'll be covering today, I'm going to start with some videos of the first commercial space flights that occurred in 2021. Next, I'll discuss the biospecimen sample collection that we performed on the Inspiration 4 crew and some of the early multi-omic insights from that study. And last, I'll touch on data accessibility and how we can make omics data from human spaceflight available to the GeneLab community. So I'm going to start us off here with a video. So this is a video from the first commercial spaceflight mission. This flew on July 11th, 2021. This mission was Unity 22. It was achieved by Virgin Galactic, which is Richard Branson's spaceflight company. And you can see here, he was actually on its first inaugural flight. The way this system worked was that there was actually a mothership plane that flew the shuttle up into the air. This shuttle detached and then rose up into space, giving the crew members a few minutes of microgravity exposure before coming back down. And that's the view from space.
Welcome to
Those two pilots were on the mothership, welcoming the crew members aboard the spaceship into space, into their suborbital flight. So this is a schematic of what that looks like. You can see the ship detaches from the mothership, climbs vertically, reaches its apogee in space, and re-enters. This next video I'm going to show you is Blue Origin's first commercial astronaut space flight. This flew July 20th, 2021. This was about a week after Richard Branson's flight. And Jeff Bezos, similar to Richard Branson, he was among the crew members on this inaugural flight. In total, Blue Origin to date has flown 6 commercial astronaut missions, and these last about 10 minutes in duration each. So Blue Origin's flights actually pass over the Kármán line, which is the internationally recognized barrier or boundary into space. The Virgin Galactic flight flies a little bit under the Kármán line, but still meets NASA's definition for the boundary of space. And here you can see the booster from the Blue Origin rocket is landing on the launch pad, or the landing pad, rather. Smooth touchdown. The capsule with the crew has separated and is in space in microgravity at this moment. I've cut out a bit of scenes in the middle here, but you'll see the capsule descending from flight with the 4 crew members aboard.
Your booster has landed.
So there is the capsule with the crew. It's about to deploy the parachutes. They've spent their few minutes in microgravity and are now returning to Earth. And what I'll show you after this are some of the reactions from crew members that have been aboard these flights. I have 2 reactions from 2 individuals you'll probably recognize. They were not on this first mission, but were on the second and third missions that Blue Origin flew. A little bit of a time lapse there. They weren't that close to the ground when the parachutes deployed, but there they are hitting the ground relatively softly. There they go. And that is Blue Origin. So those two missions together were the very first commercial astronaut space flights, each achieving around a 10-minute flight in the air. And you can see the booster and the capsule are both successfully on the ground and separate from one another. An overview of Blue Origin's flight, where you can see the capsule separation from the booster and the individual lands. And I'm going to stop talking for the reaction videos.
Done something. I mean, whatever those other guys are doing, what isn't, I don't know about that. What you have given me is the most profound experience. I'm so filled with emotion. It just happened. I hope I never recover from this. I hope that I can maintain what I feel now. I don't want to lose it.
But it's a crazy feeling, like the feeling of weightlessness, the feeling when the booster goes off, the rocket goes off, and it detaches, and you don't know what's up from down. And your body just goes like this, and you take off a seatbelt. But naturally, it feels natural to move.
You're like swimming in the air.
But if you swim, you don't go anywhere. You actually have to push off things and touch them very gently, but it feels very natural. And just see the curvature, the atmosphere, blue, the earth. It was too short. The trip was too short.
Right, like you said, it was too short. These missions with Blue Origin and Virgin Galactic are quite short in duration. All right, so the reason I show these videos, it's not just because they're landmark commercial space flights, but it's also because they give us insights into the mind of a civilian astronaut. So NASA astronauts, they do science as part of their job description. Commercial crews are under no such obligation, and their reasons for space travel can be highly variable. So William Shatner, he had a very poetic and emotional experience to spaceflight, and future astronauts may have non-scientific priorities for their space travel. It may be more for artistic endeavors or internal personal experiences. Another reason I showed this video we saw from Michael Strahan, he mentioned that these flight opportunities are very short. So there are some scientific experiments and technology tests that you can do in these short time frames. It is more consecutive time in microgravity than a parabolic flight, but it is very short duration, and you're going to get very limited time in microgravity and very limited radiation exposure due to the lower altitudes. So that said, Blue Origin does fly science on their missions. So that is an opportunity that's available despite some of these considerations. So there was one more commercial space flight in 2021, and this mission was first announced during a Super Bowl ad. I'll play this now.
This fall, Inspiration 4 launches as the first all-civilian mission to space. And you could be on board. Visit inspiration4.com for your chance to go to space.
During the Super Bowl, this was the actual launch itself. It occurred later that year in September 2021. This was down at Cape Canaveral. And we collected samples from this crew before flight and after flight, and the crew collected some of the samples themselves during the flight mission. They were up in space for 3 days, and this was the first orbital space flight. So Virgin Galactic and Blue Origin are suborbital. This one actually spent time orbiting the Earth during its time in space. So who are the crew members that were selected from that raffle? So Jared Isaacman was the first crew member. He was the financer of the mission. He's CEO of Shift 4 Payments. And he selected 3 crew members to join him. The first one was Siam Proctor. She was a teacher, a geoscientist with a PhD from Arizona. There is Haley Arsenault, a childhood cancer survivor who is now a physician assistant at St. Jude's. And there was Chris Simbrowski, who works in the aerospace industry up in the Seattle area. So this was the first all-private mission to Earth's orbit. It was a 3-day mission, and it's been regarded as a new era in human spaceflight, where we're not just sending up people anymore who have undergone years of training, who are employed by the government, who have very designated missions. These are people who get to design their own priorities, who learn how to go to space relatively quickly, and have a lot more freedom in what they do while they're up there. So for quick comparison, these are the heights that Blue Origin and Virgin Galactic achieved, 106 and about 80 kilometers for the first Blue Origin mission and the Virgin Galactic Unity 22 mission. If we look at where other things are located, we see the International Space Station is in orbit higher up. The Hubble Space Telescope is above the International Space Station. And above all of that is the orbit that Inspiration 4 went into. So this flew above the International Space Station and Hubble Telescope, increasing the amount of radiation that the crew is exposed to compared to other missions. And what is all of this building to? I think what's really exciting about this is traditionally NASA, ESA, other government agencies have been the ones pushing human exploration of space, particularly ambitious missions like a journey to Mars. And what we can do with these commercial space flights is you actually see civilians are interested in participating in the scientific and technology development to push these ambitions. So previously, what have we done with omics data? I'm guessing pretty much everyone on this call is probably familiar with the NASA twin study. This was the first omics study in a human research subject, a human astronaut. So this was a study that happened in 2015, where Scott Kelly went to the ISS for 340 days. His twin, Mark Kelly, stayed on the Earth and served as a ground control. During this time, a variety of different collection protocols were performed and analyses on different samples and measurements. So there were different biochemical and cognition tests. Of particular interest to us are the epigenomics, gene expression, immune, metabolome, microbiome, proteomics, physiological, and telomere assays. So this really laid the groundwork for how you can collect all of these multimodal types of data and perform an integrative analysis on it between a subject in flight, between pre-flight and flight, post-flight conditions, and really study how the astronaut genome is affected during spaceflight. So what were some key findings from this? This study was published back in 2019 in Science, and a general overview of what was observed during this was that there were wide swaths of changes during spaceflight. So we can see this gray block in the middle. These are summary statistics, basically summarizing the vast amounts of multi-omic data that were collected. So we can see across all these different analyte types, cognition, cytokines, metabolites, microbiome, proteomics, and telomeres, there are shifts during spaceflight caused by the microgravity, by the radiation, by the isolation. And so really what we need to do though, well, there were a bunch of follow-up studies. So it wasn't just a one and done paper. There were a bunch of different ways that people looked at this data. One thing that people found was that the immune system rapidly responded to gravity upon returning to Earth. So you can see there's different cytokines here that had a response. Specifically, blue is the flight subject, Scott Kelly, had a spike upon return to Earth after being relatively stable during flight. So we see responses like this where astronauts react suddenly upon returning to gravity. We also see changes that occur during flight itself. An example of this for telomere length, how that's longer during flight. TW was the flight twin, HR was the ground twin. We can see the subtle shift in the length of telomeres during the mid-flight time points compared to the pre-flight and post-flight. So we're seeing from these crews changes during spaceflight and also a reaction upon returning to a gravitational environment. All right, so what's the problem with this study? The biggest problem to me is that there's a small n problem. It's just a single astronaut, massive amount of data, but single astronaut, and it's just from one specific mission. So how can we go about solving the small n problem, getting an increased number of participants to strengthen some of our conclusions, that we're drawing, that we're seeing from this omics data. So as Sylvana mentioned in the introduction, we're working on something called the Space Omics and Medical Atlas, or SOMA for short. What we're doing is we're working to standardize methodology across missions. This includes both sample collection and the omics assays that we're performing. And doing this will allow us to aggregate multimodal and multi-omic data. We're also biobanking samples in this process. We're not using all of the samples that we collect. We're banking some of them so that either when new assays come out, or if we want to perform assays across multiple crews and minimize batch effects, we have samples from previous missions that we can incorporate into the study design. So for inspiration 4, what did that biospecimen sample collection look like? So this is an overview of everything that we collected. We had longitudinal data, so we collected from 3 time points pre-flight, L-92 days, L-44 and L-3. The flight was 3 days long, and we got 2 collections for a few different biospecimens during flight. Some things were more difficult to collect during flight. For example, nobody was trained to perform venipuncture in the microgravity environment. So we weren't able to collect venous blood, but we were able to collect capillary blood and saliva. We collected data post-flight as well. The soonest we had access to the crew was a little less than 1 day after they returned from their flight. We also collected data at R+45, 82, and a limited collection at R+194. So what specifically were these collections? For venous blood, we collected a variety of different tube types that allowed us to collect different components of the blood. So we collected what's called a blood RNA tube. This allows us to preserve the RNA really stably upon collection. So that gives us total RNA. We have a cell-free DNA blood collection tube, which gives us plasma that we can use for cell-free DNA sequencing. We also get a cell pellet in there that's full of various DNA that we can use for whole genome sequencing and other epigenomics work. We get a cell preparation tube, a CPT, which gives us plasma. It gives us PBMCs that we can use for single cell sequencing, and also more DNA from cell pellets. We collected 4 K2 EDTA tubes. What we do with these is we do some blood work, a complete blood count with Quest. We look at extracellular vesicles and particles, or EVPs. We do some proteomic work on that. And we can also collect additional PBMCs that we have biobanked. The last tube is a serum separator tube. This gives us serum that we can use for various cytokine panels, as well as a comprehensive metabolic panel that we run at Quest. We also get more DNA from a cell pellet. We basically save we keep those cell pellets, even though it's a bit non-standard to isolate DNA from the cell pellets of these tubes. But when you're working with spaceflight samples, you never want to throw anything away. So we have all of that still in our freezers. Some of it's been extracted to DNA, some of it's still in this cell pellet form. So we also have capillary blood. This we collect with a contact activated lancet. So you'll press the lancet, the crew will press their lancet against their fingertip. It'll form a very brief stick. And then they collect blood onto a specific card matrix called a dried blood spot card, or specifically a Wantman protein saver card. So at each time point, and they were able to do this during flight. So at each time point, crew members would basically fill each of these 5 spots on their Wantman card. So we've also collected saliva. We collected this a couple different ways. We had crude saliva, so they were spitting directly into Eppendorf tubes. And as long as you immediately store this at -80, that will be well preserved. We also had a collection device that specifically stabilized nucleotides. So this had a stabilization solution inside the tube. And we have that as well for looking at either human omics data in the saliva or microbial communities. We also got urine. We preserve this in 2 ways. First is just crude urine, just freeze the urine as it is. Second way is urine with a conditioning buffer added, specifically to help preserve nucleic acids, again, prevent any degradation. In collecting these in multiple ways, some assays that are non-omics, you don't want to add any sort of chemical preservative to it. That's why we've collected it both ways. Right, stool samples. These were actually some of the harder samples to get from the crew. So we don't have complete compliance at every single time point with this sample. Maybe it was because of how you have to collect your stool samples, but we do have some participation in this this collection. So we preserved it in two separate ways. One's with a metabolomics preservative tube, and another was with a tube specifically designed to preserve the microbiome. Okay. So we also have skin swabs. We did this in two different ways. For surfaces that were already wet, such as an oral swab or a nasal swab, they would just stick the swab right where it needed to be, move it in a circular motion, collect the microbial communities from that area. For some other locations, just to increase the amount of absorbance of the swab, they were dipped in nuclease-free water first. And then they were swabbed either behind the ear and the armpit, the underside of the forearm, the back of the neck, the belly button, the gluteal crease, the T-zone, and between the toes. And these series of swabs were also performed in flight. Right. Rather interestingly, we did get skin biopsies from the crew. This is one of, I think, the cooler omics sample types to look at, because we could actually do spatially resolved transcriptomics on these samples. That's why we wanted a tissue sample. You can look at things spatially. So we took these biopsies from the deltoid region. We did a skin swab to get the microbial community before collection. We then had a doctor perform the skin biopsy. It was a 3 millimeter in diameter biopsy. Part of that tissue was sent off for histology with the doctor. The other part of that tissue we performed spatially resolved transcriptomics on. And we got this at 2 time points. We got it about a month before they had launched, and we got it within a day after the return from the mission. All right, the last samples that we collected were environmental samples. So these were samples that were only collected in flight. Though we did collect them in some of their training capsule at Hawthorne when they were there training during a few pre-flight time points. So that data is available, but I think the data people are most interested in is the in-flight data. So there were 10 different swabs that were collected. The first was a control swab that was just held up in the air. The others were different areas around the capsule. So there's different buttons on the panels and screens in front of the crew. There's the viewing dome up where the cupola is, where you can actually get your view of space, view of the Earth. Side hatch mobility aid, which just helps you navigate around the capsule. The lid of the waste locker. Some areas around the seats, around the commode panel, different parts of the touch screen. And then there was something called a G-meter button, which it's still a bit vague to me what exactly that does, but apparently it was a button they were not supposed to press. So not the most microbial rich community on that one. All right. So from all of that, we've collected all those samples. So what data have we generated? So here's an overview of exactly which time points we collected at again. It's divided now up into a few different categories. We have this pre-flight category that was everything before flight. We have our during flight time points. Then we separated the post-flight time points into the immediately post-flight time point, and then called the other time points recovery. So this is where we're at in terms of data. We've worked with a variety of sample types at this point, though we have certainly not exhausted everything. So we have whole blood, serum, PBMCs, plasma, there's extracellular vesicles and particles, dried blood spots. We have these microbiome swabs from the oral and nasal cavities, the other swabs from the skin, as well as the biopsy, and then the microbiome swabs from the capsule. We're working on making as much of this data publicly available as possible. Some of it will be private, but still available. Like, I'll go into the process for that a bit later in the talk. But that's what this column is denoting here, is where this data will be available exactly. In the middle here, we've denoted exactly at which time points the assay was performed. So we have our assays here: whole genome sequencing, the clonal hematopoiesis assay, direct RNA-seq. That was on Oxford Nanopore. And going down the list here, metagenome, metatranscriptome, spatially resolved transcriptomics, et cetera. So we also have different profiles that we've generated. Some of these are descriptive profiles. So these are profiles that summarize a single time point, as opposed to comparative profiles, which compare gene expression or proteomics abundance across time points. So that would be when you're comparing pre-flight to post-flight or pre-flight to recovery, et cetera. Right. So there's not time to go into exactly what all the results from these assays are. So I've highlighted a few key points, a few key assays here. So we'll take a look at some of the single nuclei data that we've generated for RNA-seq and ATAC-seq. Take a look at the telomere qPCR data and the spatially resolved transcriptomics. All right. So this is what we performed for our single nuclei sequencing. We use the 10x Genomics platform. It's the Chromium, it was the multi-omic kit that does ATAC-seq plus gene expression. And we did this for 6 different time points. We have our pre-flight time points and our post-flight time points. Since we weren't able to do venipuncture during flight, we don't have in-flight for this. But we do see a lot of differences in expression and chromatin accessibility during the September post-flight time point, which was about a day after they landed. All right, so this is a very standard workflow for 10x Genomics. This is what was generated in total across all time points. We had over 150,000 cells that we profiled. These were all PBMCs. We see them clustering exactly how we would expect. We have our monocytes, our natural killer cells, our T cell populations, B cell populations, and we're seeing cluster-specific expression of the markers that we would expect for each of these cell types. Right, so what did we actually see within this data? One thing that we found was that monocytes have a much stronger epigenetic change in their ATAC-seq signal. Other cells were generally just shifting their gene expression patterns. So looking at this here, these are looking at different peaks from the ATAC-seq data. This column, or this row in the middle here from monocytes, we see that monocytes have much more differential peaks before pre-flight to the post-flight time point. When we look at gene expression data, however, it's a lot more varied. We see a lot of gene expression differences in basically every single PBMC subpopulation. So there is a difference that we're seeing between epigenomic shifts versus these gene expression shifts on a cell-by-cell type basis. The good news is all of these differential changes that we see, they tend to shift back to baseline when you look immediately post-flight to some of that long-term recovery data. So this happens for both the number of differentially expressed genes that we have per cell population, and also for the differentially accessible regions in the ATAC-seq data. We've also seen some of the chromatin remodeling reflected in the gene expression data. So specifically, we see a lot of differences in nucleosome assembly, chromatin assembly, nucleosome organization, chromatin remodeling. We also see, interestingly, DNA damage and telomere stress indicated in the data. So this leads us into doing this analysis again for telomere elongation, whether or not that occurs during a 3-day mission. So we generated this data, we were able to generate it in flight using the capillary blood collected from the Watman cards. So when we looked at the telomere length, we did this with qPCR in conjunction with Susan Bailey's lab. And what we see, we have pre-flight here done on this column here. We do see increase in telomere length during flight. It's statistically significant for one crew member. The rest of the crew members, it trends upwards. So for every single crew member, it trended upwards or was statistically significant. And we do see it decreasing during post-flight and recovery. So this was interesting because telomere elongation typically happens in conjunction with vigorous exercise when you're on Earth. For the twin study, Scott Kelly, who was on the space station, when you're up there, you have to do like 1 to 2 hours of exercise a day. So there was a little bit of a question of whether or not telomere elongation was due to the astronaut lifestyle, or whether it was truly an impact of spaceflight. So the Inspiration 4 crew is interesting because they never left the Dragon capsule, never docked with anything else. So they were confined to what is essentially a large camping tent for 3 days. They weren't really able to do any sort of exercise. And we're still seeing this effect in the telomeres, albeit a little bit more subtle than with the twin study. All right, there's my 1 minute ding. I'll go quickly through the rest of this here. So last thing I want to touch on is the spatial transcriptomic work. So there's a variety of skin pathologies that have been observed during spaceflight. We wanted to look at this on a transcriptomic level and on a skin layer level. So we worked with NanoString to do this. We profiled 4 different regions of the skin. Altogether, these are, I believe is most of our regions of interest that we profiled with the spatial system. We see clustering according to different regions of the skin. So we know each of these are distinct regions. We've seen differentially expressed genes that are unique to skin compartments. And we see that the differentially expressed genes within those skin compartments tend to be unique to that compartment and not shared across different compartments. In terms of what these differentially expressed genes are doing per skin layer, we see global differences in cross signaling and inflammatory response, whereas we see some more specific differences in DNA repair and then interferon responses. Okay, just a bit more data on what's coming soon. So some community resources in the pipeline. We're also working as sort of a supplementary resource to the Gene Labs visualization on some other data visualization software to visualize the data that we've generated here. We're working on we have excess biospecimen samples from these missions. So we're working on making those available through creating the Cornell Aerospace Medicine Biobank, or CAMBank. So you'll be able to view all the specimen samples within this biobank and submit requests for samples. These are what we're currently in the process of uploading to the Gene Lab database. We have our OSD IDs all ready to go, and we're currently uploading both raw and processed omics data. A quick statement on data availability, on accessibility versus privacy. This is a big issue with spaceflight genomics, because you can basically identify who an astronaut is from their genomic data. So there's been this trade-off between how accessible can you make the data versus how private should it be, since you can basically identify an individual's genome. And so for process data that doesn't have raw omics sequences, we lean towards making that completely publicly accessible, whereas the raw omics data, the A, T, Cs, and Gs, are going to be a bit more private. You'll have to go through just an institutional verification process similar to dbGaP, where basically there's just now a paper trail that tracks who is accessing the data and verifying that you're with an academic institution, but then the data will be accessible to those who want it. Quick statement on what is up next. There are plenty more opportunities for to work with commercial crews. There are 4 commercial space stations actively being planned. Axiom Space has already sent its first mission up to the ISS. They're eventually going to detach their own space station from the ISS and be a free flyer. Their next mission, AX2, is coming up in the upcoming months. We have Polaris Dawn, the next mission from SpaceX, coming up this summer, which will be collecting biospecimen samples from that crew as well. And this is part of the larger Polaris program, which will be a series of 3 missions, Inspiration 4, and then 3 missions, including Polaris Dawn. The first 2 of those will be, again, free flyer missions with the Dragon capsule, and the last one will be the first crewed launch, so the first launch with people in a Starship. And thank you so much to our many collaborators, and I'll open the floor for questions.
Thank you so much, Eliah. That was awesome. So we have quite a few questions for you here. Let's see how many we can get through. The first question is, did you see any biological changes in the Inspiration 4 astronauts which were surprising or different from what you had previously seen in the twin study?
So we do see a lot of individual crew member variation in gene expression data. If you start actually digging into the heat maps, looking at individual gene expression, it's not, a homogeneous profile. And so there's a lot of things that are slightly different than the twin study. And it really drives home why we need to perform this on, wide varieties of crew members really increase that N number, because these responses to spaceflight aren't homogeneous. What that means downstream when you extrapolate it to a multi-year mission, that's still very much unknown. But we can see within 3 days, people have very different responses.
Awesome. Second question is, on Inspiration 4, was the same person collecting each specimen, or did each person collect their own specimens? And if the latter, are you concerned about collection batch effect signals clouding the biological signals?
That's a good question. So on the flight, each of them did collect their own set of samples. So they were collecting their own saliva, their own swabs. And that could introduce a subtle batch effect. I'm not worried about it significantly changing the scientific results. Because ideally these tests should be rigorous enough to withstand a bit of user difference. Because I think when we're in the lab, we worry about batch effects a little bit from which person is isolating DNA, for example, but there are still these large consortiums that will aggregate data across multiple institutions, or people who download data from databases from a variety of institutions, different people doing slightly different things. And you're still able to get results from that. I think really what we need to increase our confidence in the results is just performing these over and over again so we can abstract away any individual variation.
Awesome. We got a lot of questions for you here. How do these data compare to other studies of human immune microbiome response to short periods of non-spaceflight types of stress?
So I haven't actually finished integrating all of put the Inspiration 4 data totally in context with previous studies yet. So I feel a little talking like I'd be a little bit out of my domain if I speak too much there. So I'll throw this back to you. I want to get the data uploaded as soon as possible, and whoever asked that question hopefully can answer that within the next few months.
Awesome. Next question is, will microbial specimens also be made available for request?
Yeah, so we do have some microbial data left. The microbial samples were very low biomass. We actually didn't even get a read on the qubit until we did PCR on some of the samples. And they're very noisy, but we do have remaining sample from most of those swabs. So we can make those accessible if there are additional assays that should be performed.
Awesome. What is the process used to determine which data sets should be publicly available and which should be controlled access?
Yeah. So the primary decision there is how easy it is to identify the individual and how sensitive the data is. So, I mean, the raw genomic sequence is the most sensitive data, and that's, I think, always going to be, at least for the time being, controlled access. Where what we're making accessible is more differentially expressed gene results, like from a pipeline or differentially abundant proteins. It's, in theory, you can still probably identify who some of the people are from that. But your potential to misuse that information is much less than if you had the raw omic data, assuming there's a bad actor there.
All right, we have a couple more minutes here. I think we might be able to get through a few more questions here. On Inspiration 4, besides omics, was cortisol, glucose, and or other endocrine or exocrine measurements taken from blood or saliva?
No, we don't have that. We've run some cytokine panels and some cardiovascular panels from Eve Technologies and Alamar Biosciences, but that did not include cortisol or some of those other markers.
All right, one last question here. I think we are almost at time. Are all the data from the samples shown in the slide going to be submitted to OSDR?
Yes.
All right, perfect. Maybe one last question here. What is the preservative for metabolomics that was used?
Oh, so I can tell you what company it was from. It was DNA Genotech, and they have different tubes that they use for sample collection. So we used their tubes. I'm not sure what exactly is in that proprietary formula.
Wonderful. Thank you so much. It was an absolute pleasure having you give the keynote address here. And so now we are going to move on to our next speaker.
Thank you.
And our next speaker is Begum Mathick. She is from HCA Florida Brandon Hospital, and she will be representing the Animal AWG group. And just for reference, this particular section will be speakers from the, there'll be science talks from the analysis working groups, where we'll have a 12-minute presentation, about a 3-minute Q&A. So Begum, take it away.
Thank you so much for the introduction. I'm going to share my screen now. Just one second. Can everybody see my screen?
We sure can.
Okay, perfect. Thank you. So thank you for this opportunity. Today, I will talk about our project about how spaceflight induces changes in gene expression profiles linked to insulin and estrogen. First of all, why insulin and estrogen? So both hormones are evolutionary conservative, and both hormones are very important in terms of metabolism and reproductive biology. And we already have some data about insulin resistance has been reported in astronauts, and there has been some sex differences observed, being female astronauts having better HOMA-IR values. It's also clinically important because insulin resistance is linked to polycystic ovary syndrome, type 2 diabetes, cardiovascular diseases, and fatty liver. And fatty liver, in terms of fatty liver, we also have evidence of space data showing lipid dysregulation during space flight in mice liver. On the other hand, in terms of estrogen, we have a little bit less data in terms of space biology, and it's a little bit complicated axis, like the estrogen synthesis and metabolism. So we have the hypothalamus here, you can see it. We have the pituitary and we have the gonads, so which we called it the HPG axis. Why this is important? Because due to the environment in space, like we have a lot of physiological changes, like intracranial fluid displacement. It can involve like pituitary gland. It could affect maybe FSH and LH secretions here. We also have some changes in circadian rhythm, which is also closely related to reproductive biology. There is also effects of microgravity and fluid shift. When we were talking about the fluid shifting upwards, also the lower extremity fluid is going to be decreased. So we don't have data much about indeed the pelvic organ blood supply. There is another important thing is that we all know ovaries are radiosensitive. We now have this data as a clinician, but also there is a space radiation. So the radiation is different. So that's why there are possibilities at the different levels that may affect estrogen synthesis, metabolism, or signaling. And also both hormones are interact each other. Like you can see, estrogen improves insulin sensitivity in animal models. Estrogen receptor activation improves hepatic steatosis. And when you have knockout mouse models too, you have hepatic insulin resistance. And in terms of human data, we know that during the postmenopausal transition and postmenopausal time, there could be alteration and dysregulation in lipid profiles of the patients and more prone to insulin resistance. So what are the possible mechanisms of insulin resistance during space flight? So it could be due to altered lipid metabolism, and it can create a vicious cycle between both of them. And we have data published about this one. It could be due to mitochondrial dysfunction and endoplasmic reticulum stress. We also have proof of mitochondrial dysfunction during spaceflight. It could be also due to circadian rhythm changes. Another issue in space biology is the immune cell dysfunction, but this is the area that we don't know much, how the estrogen signaling is changing, and if so, how it's going to be related to insulin resistance. So this is our team who made this study possible. So I just wanted to show our team before we go talk about more about the study. So this is our graphical abstract. To understand those two hormones, what did we do? First of all, we utilized the NCBI gene list for genes linked to insulin signaling, genes linked to estrogen signaling, and also a separate gene list for the insulin resistance. Then we have the female mice here, which is from RR1 mission. It was a 37 days mission at ISS, and the mice euthanized at ISS. And we have different organs from the same mice cohort. So you can see even different sets of muscles here: liver, eye, adrenal gland, and a kidney. We have the bulk RNA-seq data from this one processed by GeneLab. We also have some human data, astronaut data from JAXA and Inspiration4. I'm going to start to talk about first the mice data, then I will continue to talk about human data. When we first analyze the female mice data, we look at all the organs that utilize the RNA-seq data, and we notice that spaceflight has a global impact on differentially expressed genes linked to insulin and estrogen signaling, and it was like across different organs. Because we have a lot of findings, I need to focus one of the most important ones, and I will focus on liver here. When you look at this column, you can see it for both insulin and estrogen linked genes, they were downregulated in the liver. And in the next slide, I will explain why this is important. We also performed gene set enrichment analysis, and I will specifically talk about liver in this one again. But you can see also different levels of insulin and estrogen gene set enrichments across tissues. This site is mostly muscle, and this site is the other organs. But when we look at the liver again, you can see most of the gene sets related to insulin and estrogen pathways, they were negatively enriched in the liver data sets. And I can give you a few examples. One of them is the insulin receptor signaling cascade. Another one is response to insulin. And another one is the estrogen signaling too. We also performed engineering pathway analysis, and we specifically look at the genes, differentially expressed genes related to insulin signaling in the mice liver. And when we look at them, you can see those are the downregulated genes in the insulin signaling pathway, and their downregulation related or associated with the increased activity of hepatic steatosis and insulin resistance. Indeed, this was interesting to see because we have a publication already from the RR1 mission showing also histologically evidence of lipid accumulation in the same mice cohort. So when we are moving forward with the human data, I just need to move this panel a little bit on here maybe. We are going to look now the expression of insulin-linked genes and how they change pre-flight and post-flight. So we have the gene sets from NCBI again. Then we have the plasma cell-free RNA-seq data from the astronauts. And this heat map shows the insulin signaling upregulated genes during post-flight. And this one is the differentially expressed genes downregulated during post-flight. And I want to show you, we cannot go all over the genes separately, but I just want to point out a few interesting things in this slide and the next two slides. So first of all, interestingly, when you look at the changes during flight and post-flight, some of the genes change throughout the whole post-flight duration, regardless of when you're back 3 days, 1 month, 2 months, or 4 months. But some of the genes have a specific pattern, initial upregulation, then going back to baseline. So this was interesting observation, and we try to still working on which group is having like a different expression profile. And I can give you a few examples also in this one. In the post-flight, you can see estrogen receptor 1 here, and this is like AKT1 here. Especially AKT1 is upregulated throughout the post-flight, all the periods and time frames. And this is very important downstream signaling of insulin. And on the other hand, when we look at other important changes, this is a glucose-3 transporter gene encoding GLUT3. So this was downregulated during the post-flight period too. Then we have this heat map, and we plotted the pathways based on the changes on these heat maps. And we see some enrichment of specific pathways, and with the differentially expressed genes in the insulin signaling, the significant or enriched pathways were, one of the top one is the cholesterol metabolism, interestingly. But also you can see, even though we are looking at the genes related to insulin signaling, you can see the enrichment of estrogen signaling pathway here too. And some of the enriched pathways are also interestingly associated with infectious diseases. So this slide is a similar setting again, but this time we look at the estrogen signaling genes and how the pathways change post-flight, compare upregulated post-flight and downregulated post-flight. And I will give you a few examples again, because we cannot go all over each gene, but there are some interesting ones. One of them is the circadian rhythm. It's upregulated during the post-flight period, especially in the early time frames. And another one is cytochrome 17A1. This is a gene encodes the stereogenetic enzymes. This was upregulated throughout the post-flight period. I found this one is interesting too, follistatin, because follistatin is important in terms of reproductive biology, but it's also important for muscle mass as well. And this is, again, we look at the same heat map and we perform the pathway analysis, but this time we utilize the insulin-resistant linked gene sets. Then we observe some changes as well. One of the most important here I would like to highlight is this CRP. This gene encodes the C-reactive protein. This one is upon return, the first 3 days, this time frame, it was upregulated. But then it's changed and it was upregulated again towards to the end. So I think this is important for long-term follow-ups because CRP itself clinically is also associated with insulin resistance as well. And another important one is sex hormone binding globulin. You can see the regulation of this gene being upregulated or returned to baseline at different time intervals. This is also important because it can affect the free concentration of the circulating hormones. And then you can see the insulin resistance as a here again as an enriched pathway. We also further look at the other human missions and tissues, and we observe some sex differences in this one. One of the important one is this. We have the single cell gene expressions of PBMCs here from the Inspiration4 data. When we look at, especially in the B cells, and especially under estrogen signaling gene sets, you can see the ratio female differentially expressed genes versus the ratio male differentially expressed genes. And especially in the B cells, the downregulated genes of B cells were higher in females. So we saw some sex differences in terms of subcellular cell types here. We also have the skin biopsy samples from this mission. And depending on the depth of the biopsy, there is the categorization of the compartments. And then we saw that the region-specific sex differences observed when we look at the different compartments. But especially this was more prone for the vascular region in terms of insulin resistance. It was more enriched in females than males. And as a conclusion, we observed some changes, genes associated with insulin and estrogen signaling, and they were altered during and after spaceflight in both rodents and people across various tissues, because we analyzed different tissues in both rodents and human. Some of them upregulated, some of them downregulated, but there is definitely a change and alteration during spaceflight. And in the last slides also, we discussed about this response may be different in men and women. And why this is important, because as I mentioned in the first slide, insulin and estrogen dysregulation can cause disease states like cardiovascular disease, diabetes, and osteoporosis, and like fatty liver. And that's why future studies should target more reproductive organs and endocrine tissues. Thank you again for our team, and special thanks to Chris Mason and his team for Inspiration for Data, and special thanks to Masafumi Muratane and his team for JAXA Data, and also Afshin Beheshti for introducing me to GeneLab. Thank you.
Thank you so much, Begum. That was amazing. So we have a few questions for you here. First one is, can you please speak about the relevance of your findings for terrestrial diseases such as polycystic ovarian syndrome and non-alcoholic fatty liver disease?
Perfect. Thank you so much. So basically, based on the rodent data, I can say that we definitely can, we have an evidence of fatty liver disease, but also like we don't have a liver from human. But I think future studies is going to give us more information about the lipid profiles if we have a long-term follow-up and different sequential timing sampling during the flights as well. And in terms of polycystic ovarian syndrome, that's also my question too. Like if you have it and if you're going to develop an insulin resistant, is it going to make it worse when you're going to a deep space journey for a longer time?
So that kind of leads into the next question is, in your opinion, what do you think is the most likely cause for the hormonal dysregulation you are observing? Is it radiation? Is it microgravity, overall stress, or some combination?
So we look at the gene level. Also, it's I need to like have more data about in terms of blood sampling and hormone levels. But my hypothesis and our group, when we discuss it, it could be overall effect. That's why I showed the HPG axis. There are multiple levels that estrogen signaling or estrogen metabolism can be affected. And I can talk about more to like each level or like.
Thank you so much, Begum. We are out of time. I really appreciate you giving us your time in this wonderful presentation. We do have to move on to the next speaker. All right, so our next speaker is Andres Caicedo. He is from the Universidad de San Francisco de Quito, and he will be representing the multi-omics AWG. Andres, go ahead, take it away. Andres, you're muted.
Yes. Thank you very much. Thank you very much to everyone. I'm setting up my presentation at the moment. I will be with you in a second. Perfect. Okay, I hope that everybody's seeing it.
Yes.
Yeah. Perfect. Thank you very much for this opportunity. It's a pleasure to be here. I'm going to talk to you about our project, the identification of key genes and their altered pathways in sarcopenia as potential biomarkers for muscle loss in astronauts. Okay, so I would like to introduce you our group here in Ecuador and the Universidad of San Francisco, which is composed by Veronica, Alison, Yvonne, Juan Esteban, and myself, which we are very interested in human health, and we use cell biology and bioinformatics and big net analysis tools in order to propose new biomarkers and also new therapeutic use to be further studied. And I would like to also thanks to all the people that were involved in this work, especially from the AWG international team. I'd like to thank Jiwon, Jangkyung, Eliah, Gino, Andrea, Christopher Mason, and Afshin Beheshti, who really support us and provide all the help in order to develop this project further. So I'd like to bring your attention to something that is happening, that after 60 years of space research, maintaining the health of astronauts during the space and after they're returning to Earth is still a challenge due to the fact that we still don't know much about what is happening to our biological systems when they are outside there and when they are especially returning. So finding new ways to preserve their health is extremely important. Astronauts suffer from microgravity, from ionizing radiation. They suffer from many sources of stress, like also isolation, mental exhaustion, and everything, and that can damage their biology and also damage their homeostasis. So, but especially by the lack of gravity, astronauts suffer from muscle loss and the reduction of bone density. Interestingly, this muscle loss and reduction of bone density has been linked with the weakness observed in sarcopenic patients here on Earth. So the lack of gravity induces this loss of muscle mass, but also old age and the lack of activity and also the lack of proteins is harming the old population and make them then lose their muscle. And this loss of muscle is something that we really need to take care of, because this led to an accelerated biological decline and also frailty, meaning that if you lack the capacity to perform activities, you will also increase your or decrease your capacity to do activities that will help you to maintain a good health. So this is very important. So regarding this part, new biomarkers of muscle loss and also understanding the biology of each of the individuals that go to the state of our astronauts that go there will be very important in order to suggest personalized pharmacological interventions that can help them to preserve their muscle mass and to have a safer return to Earth and also an adaptation to gravity. So the first challenge on this project was to identify the key genes in sarcopenia that were related to muscle loss, and we wanted to do this by the help of Earth's precision medicine, in which we found three data sets who were coming from different parts of the world, from Jamaica, from Hertfordshire, and Singapore, that we all studied because they were muscle biopsies from healthy and sarcopenic patients, and we used 180 samples from RNA-seq, in which we analyzed the individual protective profiles of these genes and obtained the 687 differentially expressed genes that were correlated with sarcopenia. We performed a co-expression network analysis from which we obtained gene models with the highest correlation with sarcopenia. Thereafter, we perform other analysis, and in this heat map, we present the expression values of these DEX with an average accuracy score to be linked to sarcopenia higher than a 75%. So this is a very good pool of genes that we need to reduce. And in order to reduce them to have a more focused gene set, we perform a gene ontology analysis from the Brown model, then the overlap with the differentially expressed genes with different levels of significance, and the network analysis using cytoscape from the Brown genes. And we obtained 21 key genes that were highly significantly correlated with the disease, and they have, among the different profiles, very important biological pathways that are deregulated in these samples. So we analyzed the mean accuracy score of these genes related to sarcopenia and found different responses, for example, from protein ubiquitination to cellular response to stress, obviously that were linked to the sarcopenic phenotype. Later after, we wanted to observe the expression of these key genes across the patients with and without sarcopenia, clustering them via key means and the Euclidean distance method. From this part, we were able to observe that the genes that we were able to analyze and to identify correlate with the healthy and sarcopenic arrangements that we observe, for example, in this heat map. Thereafter, by identifying these key genes, we wanted to enrich and to observe the key cellular functions that were perturbed on these ones, and observe, for example, that in ADPH activity, fat, proton channel expression, melanocortin, and glutamate activity were perturbed. So this brought us attention to many interesting metabolic pathways inside the mitochondria and also outside mitochondria. So after finding these 21 key genes, we wanted to test them now in the samples that were coming from space, from the space flights, from the data available from AWG. And this represents a very interesting advancement, because we are comparing the data that we obtained from patients here on Earth and comparing it and validating it from the second space age data. So we wanted to compare from different samples. One of them is UVEC cells, which are, well, isolated cells that spend time in space, plasma, cell-free RNA coming from JAXA, and skin and blood samples coming from Inspiration4. What we observe, interestingly, is that when you are using single cells, and these single cells are not in a systemic response to space, we were able to observe that no deregulated genes were found. So this is very, very interesting, and also, well, very comprehensive, because muscle loss is a systemic response that even involves the activity of the immune system. So this is something that very interesting, and remarks the importance of using more complex tissue samples than rather only cells. Later after, we made an analysis of the gene expression from JAXA using the cell-free epigenomes. Like we use these data cells coming from a sample of plasma, analyze the cell-free RNA from the different time zones of the mission, the pre-flight, the flight, and the post-flight. And we were able to observe that these 21 genes were differently clustered in these samples. So this give us the insights that the genes that were associated with sarcopenia were actually moving in this interesting blood sample, which brings us to the idea of what could be used as a liquid biopsy in order to find markers that can be used to understand the physiological response of our astronauts in space. Then after we used and validated these key genes across the different skin compartments, as was mentioned before, we were able to observe that the genes that we identified were differently deregulated in the different layers of the skin that were analyzed. And this is very interesting, as bring us some ideas that the complexity of a tissue is highly responsive to the different stresses that are suffered during the space missions, and that after flight can be deregulated, and it could be very important to know how to maintain them at good levels after the astronaut came to Earth. Then as it was important to observe the different data sets and also to validate the key genes that we were analyzing, we observed the dynamics of the expression of the blood cells. And we were able to analyze our genes in B cells, CD4 and CD14 cells, but especially in PBMCs, because, well, this is a very complex population of cells, but they reflect how our genes are, the identified genes in sarcopenia may be acting. And interestingly, we were able to observe that during pre-flight and post-flight, the identified genes were deregulated, and some of them were common to most of the cells that we were able to observe. And this comes to this heat map, in which the genes that we identified, 14 of the 21 more specifically, were able to be highly represented in most of the samples that we observe. For example, in the T cells and the PBMCs, and that's from the skin. And we observed that, for example, POMC was very well represented in all the samples. So this could be a very interesting and key biomarker of the possibility of an astronaut to develop sarcopenia, and even from people on Earth to develop this condition. And what is extremely important as well is to exploit the data that is available and to propose and to suggest new pharmacological interventions that could help us to maintain the genes that are regulated, or the genes that are trying to maintain the health of a tissue in space by pharmacological interventions. So based on the chemical interaction of these key genes, using a comparative toxicogenomics database, we were able to observe that key genes, for example, from C, NOX1, SHD, and GLS2, were able to be regulated by some pharmacological interventions that are already in clinical trials or in the market. And this is very important, as pharmacological interventions and suggested drugs could be used to help to preserve the muscle and decrease the stress response from the astronauts in space. So the take-home message, thank you. The take-home message could be that based on the analysis that we performed, that using Earth's precision medicine, we were able to identify 21 key genes that were highly related to sarcopenia, and that these key genes, after further analysis of the tissue samples that were studied, can be used to have a good biomarker list that can help us to predict the predisposition of an astronaut to maybe lose muscle mass. Maybe they will need to exercise a little more, to have supplements that can help them to prevent them, and also to help them to have a safer return. So these 21 identified genes and the analysis that we perform will help us to also to choose the best sample to take to have this ability to analyze the gene expression in these astronauts, and to propose personalized pharmacological interventions that could make space travel safer, and also to help astronauts to arrive to Earth with a better capacity to pass over this stress response that's been seen in the first days. So thank you very much to everyone. Thank you very much for this opportunity, and to everybody on the team that provide the analysis and the work of this study. Thank you very much.
Thank you, Andres. That was an awesome presentation. We do have some questions for you here. Let's see how many we can cover, since we are running a little bit late. But question number one is, do you think that your experimental approach using terrestrial precision medicine and multiple data types could be applied to other diseases that are at risk in spaceflight beyond sarcopenia?
I think that is very important. Thank you for this question and comment. I think that yes, by using data from precision medicine here on Earth, we will be able to, for example, have more ends, for example, than the astronauts that went to space, and is a way to shortcut the lack, for example, of samples, and to target some key genes of some diseases that are present here on Earth, but also out there in space. So this is very interesting, and will be a very good point to try to find significance of the biological response that we are dealing when we go to space, of course.
Wonderful. Thank you, Andres. I'm sorry we are running a little late, so we do have to move on to the next speaker, but thank you so much for your time.
It's a pleasure. Thanks to you. Bye.
Wonderful. And our next speaker is Stephen Lanton from the University of Florida, and he will be representing the plant AWG. So go ahead, Stephen, take it away.
Great. Let's see. Can you see this?
Yes, we can.
Great. So hi, everyone. My name is Stephen Lanton. I'm a PhD student and NASA Space Technology graduate researcher at the University of Florida. I'm giving the plant AWG science talk on a hyperspectral imaging pipeline for controlled environment and space agriculture. So in contrast with a lot of the fundamental space biology research that I think this group works on, my work sort of more revolves around space technology itself. And so one of the core questions when we're trying to motivate space technology research is, what is still needed to be able to live in deep space? And, we have the NASA technology taxonomy that sort of formalized these needs. And one of these needs is that we need to be able to store, prepare, and consume food out in the deep space environment. So this is nice and fine, and we've been feeding space station crews for decades in low Earth orbit. But I think we're going to need to rethink how we feed crews in deep space, as we go to the moon, as we go to Mars, for a few reasons. And the first of these is that there's a lack of nutritional stability in prepackaged space food, not to mention the cost of resupply. Second is that as we have more deep space missions, crew time is going to be reallocated to more maintenance type tasks. And so, there's less time for them to perform experiments, to actually, grow food in deep space. And the third is that crops are usually subject to contamination when they're grown by crew. So, what do we do about all these sorts of things? Well, we can grow plants in space, but we can do so using a higher degree of automation. And so what this does is it actually develops a sort of sub-need for these autonomous systems to be able to, one, monitor plant health non-destructively, and two, we need to be able to determine the nutritional content at harvest. And so one way that we can do this is through hyperspectral imaging. And so if you're not familiar with this, it's basically taking pictures of plant canopies to figure out how well they're growing and how nutritious they are. But instead of taking pictures with just any camera and getting back 3 wavebands of data, so we get our typical red, green, and blue, what a hyperspectral camera does is it takes photos in hundreds of wavebands with about a 1 to 2 nanometer resolution. And so with this really tight resolution, that actually allows us to tie image data directly to plant biochemistry. And so the way that this works is, you start with some light source, it shines light down on a plant, and the photons from this light, of different wavelengths, they're differentially reflected based off of the plant composition, right? And so some of that light hits the hyperspectral sensor, the signal is transduced. And so now we have something called a hyperspectral data cube, where we have hyperspectral data in 2 spatial dimensions, right? So we have our X and our Y, but we also have another spectral dimension, this lambda dimension right here. And so we can think about, once we clean up our hyperspectral data, make sure it's all post-processed correctly, we can think about using these spectra to develop really good prediction models to predict, plant health and nutrient content non-destructively. And so, a lot of research has been done actually in field agriculture using different types of imaging techniques. And what people have found over the years is that there's a few imaging regions that are most relevant to plant health. And so we look at the visible and near infrared for things like chlorophylls, carotenoids, lots of the vitamins that are actually present in plants. We can start to predict their content, but we also look at the shortwave infrared to look at things like stressors, like abiotic stressors, like water stress. And so, so how do we go about building these prediction models? And it turns out there's this sort of standardized data collection scheme for this. So what we do is we start with a randomized complete block design experiment, with varying degrees of treatments. We let these plants acclimate to these treatments and then image them hyperspectrally. And then what happens is those same plants that we image, we also perform destructive analysis on them to get ground truth data. So what happens is, we have our hyperspectral data, we have our ground truth data. This can be usually collected through HPLC or different types of destructive techniques. Once we have these data sets, we can actually use the hyperspectral wavebands as predictors, and our destructive methods, the values we get from our destructive methods as our response. And then we can start aggregating the data and running and building prediction models through these sorts of data sets. So it's really, really valuable stuff. And we've actually started to build models for things like nitrogen, phosphorus, potassium, lots of different plant constituents. And so, you can think about actually training these models and then applying them to new images of plants that haven't been exposed or weren't used for the training data sets. So here we have this image of an anthocyanin model that's overlaid on top of two different cultivars of lettuce. So one of them is just a regular, green Bib lettuce, and the other one is a red romaine lettuce that's actually known to sort of upregulate the anthocyanin content in these plants. And what you find is that with these anthocyanin models, they can actually start predicting the content in the plants sort of non-destructively and, autonomously. So, when you properly train and validate these models, they're a really good prediction tool to actually apply to any sort of hyperspectral image of plants. And so what happens is, we can build these models, but at the same time, we run into challenges when we actually go to apply them. And there's a few reasons for why this is the case. So first issue is that, there's really good hyperspectral image sensors, but they're really, really expensive. And so one solution that we came up with this is we do some sort of a bare bones build, right? So that can collect the same data for a lot cheaper. And so this was actually a project I started as a visiting technologist last summer at the Kennedy Space Center. Really happy to continue this with some undergrads at the University of Florida. And we've managed to build up a rail-based spectrometer system that you see here. And so you can see that it's mounted in our chamber, it's collecting data autonomously. Really, really neat stuff. But one thing is that, it's one thing to build up the hardware, but if you don't have the proprietary software to go with it, that actually does the data collection and cleaning, it's sort of useless too. So what I've done is I've actually rewrote a lot of the radiometric calibration software in open source Python to be able to sort of make this available to more people. And so what we find actually is that when we compare the spectra collected from really expensive hyperspectral data collection software and hardware system and compare it to the one that we built, we actually find that the spectra actually line up pretty well. And so we think that this could be a really nice drop-in replacement for something that's a lot cheaper, right? Get a lot of very similar data for something that's about 1/10 of the cost. So we run into another issue when we're actually trying to apply these hyperspectral models, and that is that in order for a lot of these models to be useful, we have to be able to characterize the hyperspectral response for many factors and their interactive effects. So I'm talking about things like, species, cultivars, phenological stage, and all the different stresses and nutrients. So, as you can see, these factorials start building up really, really quickly, and a lot of experiments need to be performed, right? So there's really this roadblock in the sense that we need to be able to collect a lot of data in order to parameterize this entire grow space. So our solution here is that, why not make this pipeline economically and informationally available in order to accelerate the data collection process for these, hyperspectral models to be useful. And so, luckily we have the NASA Gene Lab and the OSDR that's really working really hard to curate a lot of these data analysis pipelines. And one really cool thing that I found with the analysis working groups is that one of their main goals actually is to develop a canonical data analysis pipeline. So I think there's a lot of really good synergistic effects here. And just really quickly wanted to give a shout out to the AI and machine learning, AWG. And I just learned about the concept of self-driving labs through them. I think setting up something like a hyperspectral data collection system in this way would be a great way to collect data in a large parameter space, such as this one. So now, once we start to accumulate data to sort of more fully characterize the parameter space, we can start to develop digital twins to enable real-time prediction and control of plant growth all the way from sowing to harvest. So this is really the end stage where we have something that can actually grow plants autonomously, where our crew doesn't really have to worry about growing the plants. All they need to do is just plant the seeds, come back 28 days later or however many days, and be able to harvest their plants. And so the way that this works is, let's say we have an objective to grow some type of lettuce. Outrageous is the one that we grow in the International Space Station usually, with X amount of micrograms per gram of lutein. using the systems that we have already available to us? Well, it's sort of an open-ended question of how we should vary the light recipe, nutrient solution concentrations, all these sorts of parameters over time to actually achieve this result, while also accounting for system disturbances that might arise in the spaceflight or, surface-based environments. So first, we start, with our physical system with a lot of different parameters that we can change in order to upregulate or downregulate different types of nutrients and, different constituents in the plants. So we have our variable spectrum lights, we have temperature that we can modify, relative humidity, CO2 levels, and we also have our nutrient solution concentrations. Now, we actually pair this with a virtual model. So this is ideally pre-built from first principles. You'd have to develop these models of what an ideal plant would look like. These can be functional, structural, or both. And basically what you do is you sort of start running these in real time. And so you sort of pair the physical system with the virtual model in real time to be able to generate a lot of these insights that we can get from digital twins, such as, prediction and control of future behavior. Great, thank you. So once we can think about, developing a system like this, we run it very regularly. So, we run our data collection for every sampling time step. And what happens is we can actually, first the sensor network updates with real-time data, right? So it'll update the virtual model. Then what will happen is, you have your physical systems parameters that are compared to the ideal model in this sort of process control scheme that, we're developing currently. The process control scheme would actually produce new control set points. And then these set points would actually be used to inform the actuators that would actually make those adjustments in lieu of astronauts actually, adjusting the system themselves in order to actually, satisfy the need to be able to grow plants autonomously and non-destructively, and also be able to do it in a way that, determines their nutritional content at harvest. So I think, with this sort of paradigm, we can start building robustness and automation into our space plant production systems in order to make them more reliable for deep space missions. So with that, I think I'll wrap up. Thanks very much for listening. I'd like to thank my advisors, Aditya Singh, Melanie Carell at the University of Florida, my NASA mentor at KSC, Dr. Joya Massa, bunch of graduate students and external collaborators, as well as undergrads who really make a lot of this research possible. Thanks.
Thank you so much, Stephen. Awesome presentation. We do have some questions for you here. Are the videos of plant growth useful for plant health biomonitoring and predictions?
I'm sorry, did you say videos?
Yes.
Yeah.
Videos
So it depends. So if you're talking about videos, usually I think they're more RGB based. And so while they will give you some information on how the plants are doing, you could really think about maybe doing like a pseudo NDVI sort of metric to figure out what's going on inside the plants. I think without having hyperspectral data, I think that's a little bit tricky. But you can look at things like biomass and other things that you wouldn't be able to detect with just a point sensor.
All right. Next question is, cool system. Do you think it could be used to analyze the impact of soil microbiome changes to plant growth as well?
Yeah, so there's a lot of actually research being done at the University of Florida looking at different types of, soil-based approaches to growing plants. For those sorts of things, I think maybe not so much hyperspectral. You could take a look at it, but it also would depend on the constituents of the soil. So you basically would match up your imaging tools to detect different changes, in the soil. And I'm not sure if that's in the visible or near infrared spectrum, but you can think about adapting different camera systems for your needs.
Awesome, thank you, Stephen. We are a little behind, so we do need to move on to the next speaker, but thank you so much for your time.
Thanks.
All right, and our next speaker today is George Myers from Michigan State University, and he will be representing the Artificial Intelligence Machine Learning AWG. Take it away, George.
Great, thanks everyone, and thanks for the opportunity to talk and for the great talks that we have heard this afternoon. I'm gonna talk to you about detection of time-resolved. I think my screen sharing just stopped. Sorry about that. Let's try this again. Can you guys see the slides?
Looks good.
Okay, great, thank you. So I'm going to talk about detection of time-resolved molecular changes in astronauts during long-duration spaceflight. We should take a step back and think about current medical practice when we're thinking about how to tackle space medicine. So in current medical practice, we know has limitations. So typically when symptoms present, there's a clinical evaluation that is actually performed typically by a physician at a hospital, and a set of limited measurements and tests are going to get carried out. And these are typically followed by a waiting period for the test results, and there's going to be a probable diagnostic, probably not specific enough. Testing will have to be repeated, treatments adjusted, and this is a lengthy process, and it can be detrimental to a patient's health. And we need ways to actually improve healthcare to enable early and precise diagnostics and treatment. Now we take this model and we're talking about implementing space medicine, which presents additional challenges, basically. And so diagnosis has to be done on site. This is especially important for any deep space missions in the future, and relying on Earth for remote diagnostics would be problematic. Furthermore, sample processing will have to be done in space, since there are huge logistic issues with returning samples for processing to Earth, as we have already found out for low Earth orbit missions. So time here is key. Monitoring has to be performed over time to detect deviations from some health baseline. And then there's going to be some time to diagnosis, and treatment has to be as brief as possible. So in terms of monitoring, again, I focus on time. Time is essential. So there are various risks that are associated with space travel, and these manifest over time through a period of change. So there's changing gravitational fields that can affect the body. We can think about the changes leaving Earth to weightlessness when you're in orbit or beyond Earth, or reduced gravity. And that's going to happen either on the moon or on Mars. There's going to be problems with isolation and confinement over time for months, and this can have detrimental physiological as well as psychological effects. The astronauts are going to be in a spaceship in a closed, hostile environment, and the ecosystem changes again over time. The immune system can be affected. Obviously, radiation exposure is one of the greatest risks without having the Earth's protective magnetic field there, and this needs to be monitored over time, including for accumulation of effects. And finally, being away from Earth, that's a big problem, right? Not just for communication, but also for getting supplies and diagnosis and treatment. Now, we have great technological ability now, and we're still building on this to monitor thousands of measurements over time. Let's call all these measurements generalized omics. This includes all the traditional measurements that we can think of, for example, whatever we get using blood panels. But now we've been expanding these to include profiling of metabolites, looking at the cellular components, different molecular measurements, including transcriptomes and proteomics that we've heard about, and also increasingly using this mobile device data, and that includes looking at ECGs and EEGs and more. We're adding more and more measurements all the time. So given that we have all this big health data that we're monitoring, we need to develop new algorithms to monitor changes from individualized, personalized health baselines. Medicine is an individualized process, right? So we need to figure out changes at the individual level compared to their baseline health. We want to implement this kind of personalized medicine in space and on Earth also, of course. And we want to be able to monitor deviations and detect potentially adverse health events relative to each astronaut's on baseline while they're in space. That's the main idea in terms of our framework. So what we're trying to do is we're trying to use machine learning methods and artificial intelligence in space health monitoring. And we want to not only automate detecting any events and potentially adverse health events, but also interpreting these and providing actionable information for autonomous healthcare in space. So we've been carrying out a study that monitors multiple molecular measurements for astronauts, and we've been utilizing time series data that have been collected from 2 studies, a nutrition and an immune function study. That's SMO 015 and 016E. And this data spans 32 missions, basically. This is the largest individual astronaut time monitoring to date, as far as we know. We've consented 38 astronauts. There were 47 available, and we analyzed data from 27 of them that were from long duration missions to the International Space Station and stays there. And those were approximately 165 days in the mean. So these data include blood and urine and metabolites, and also vitals and immune information reports. So like I was saying, our goals, one of the goals is to detect potentially adverse health events and classify these. So to do that, we've created algorithms, including this Iomica framework that we have. This is software Mathiomica and Piomica, and we've taken a 2-step approach. In the first approach, we establish baseline characteristics for each astronaut and detect baseline deviations for every individual. And then in the second step, we compare time result changes across individuals and try to find a common cause of any potential adverse events or any common signatures of those. So here's a little summary of the implementation. I won't go into too much detail, but the idea is that for each astronaut's omics time series, we analyze these using spectral methods, so methods based on Fourier transform analysis. And we're using periodograms and autocorrelations. Those are correlations of signals with delayed versions of themselves by some kind of lag. And using that information allow us to classify the different temporal patterns, changes over time in these signals, and identify which signals show statistically significant non-baseline behavior. Then we separate these signals based on how similar their trends in time are. And so this classification enables us to identify potentially adverse medical events and their onset times. Once we do that, then in part 2, the different omics from all the astronauts are integrated together using network methodology. And that way we can compare and identify measurements having similar profiles across the astronauts, as well as groups of astronauts that would have similar changes over time. And to do this, we developed Piomica. This is an open source Python package. All the source code and full documentation are freely available online. And we're building on this and adding new information. And we used Piomica to calculate the periodograms and autocorrelations for each measurement time series from the data that we had for each of our astronauts. Then we use these spectral analysis results to identify and categorize the trends in time and summarize these using graph theory with something called a visibility graph that will show us, give us a representation of the time points of potentially adverse events and then changes that happen over time. And then we use community detection to identify astronauts that show similar behavior and assess the biological and physiological significance. In doing this, we have to address challenges that we all face with our data, especially for human astronaut data. We have thousands of time series for each person that we have to analyze for statistical significance. We're dealing with different scales and normalizations for all of these different signals. There's different time frames for the measurements and different sampling rates that needs to be taken into account. There were missing data and the data were unevenly sampled, and all this info needed to be integrated and interpreted biologically for medical relevance. So we think we've addressed a lot of these points, at least, with our methods, one implementation. There are many methods to follow. Here, I'm going to show you some examples. You're seeing example output from the individual analysis of 2 of our astronauts. In both of the figures, you see detected trends in these heat maps. You see this circular graphical summary, that's the visibility graph in the middle, and then a summary of the corresponding signal autocorrelations on the right. If we concentrate on the heat maps, you see time shown on the horizontal x-axis. There's a common time binning across the astronaut analysis. These are median times because the astronauts have different duration stays in space, and we don't want to share those for to avoid attributability and protect the privacy of the astronauts. The first 2 points here are before flight. The next 3 time points are during flight, and then we have return to Earth. Then the last 2 points are 1 and 2 months after returning from Earth, respectively. So if we look on the left, let's call this astronaut 1, and you see there's a set of measurements where intensity is lower in space, and then it recovers when you come back to Earth. And then there's another set where intensity increased once the astronaut was in space, but then it came back to lower levels after coming back to Earth. That's recovery. On the different example, if we look at astronaut 6 on the right, we see there's a change in intensity of several measurements after going to space, but these effects seem to persist after they come back to Earth, at least for 2 months after they're back. That's the duration of the study. And that means there could be significant long-term health implications that the astronaut's health does not return to the pre-flight baseline. So for the astronauts' individual classifications, we can obtain all the signals, and that shows all these kinds of patterns. And then we ask, are they significant? And then we can see across many astronauts that there are many signals that are significant. Using this bottom-up approach, we can accumulate them and count how many we have. And so there are multiple measurements that show that there are systemic changes. I'm showing some top ones here, and they're associated with bone resorption or myofibril or protein breakdown, acid metabolism, kidney function, immune alterations, and immunosuppression. Don't have too much time, so I'm gonna just show you some examples. What you're seeing here in this example is changes in pyridinoline across subjects, and you see this global increase. And so pyridinoline seems to change during flight, go high, and stay high after the return from space. And these are clear. This is a marker of bone resorption. This is observed in patients with metabolic bone disease. This is a major concern for bone loss among astronauts. Another example here is sodium urine. You see fluctuations, and then you see retention and decrease upon return to Earth. And that's the effects of microgravity on body fluid balance. You can see similar effects in zinc and serum. In phosphorus, changes that happen over time in the astronaut, decreases that are not recoverable after returning to Earth. And lots of changes, including in immune cells. You can see here decreases in monocyte percentage counts. We notice that this is across the board. A decrease in the monocyte counts, similar changes in CD8 positive naive cells, but this return a little bit to normal, but that return is modest. This decrease shows that there's changes in T cell maturation. So in conjunction with the monocyte results, this means like there's probably a decrease in the relative proportion of immune cells. And this could be linked to viral reactivation, for example, latent herpes virus reactivation. And we see something similar in the lymphocytes. So we can count all this information across all the astronauts, and we get cumulative counts, and we see the patterns across all of these. And we also have different conditions that the astronauts experience. So sleep disorders, fatigue, space motion sickness, and the like, insomnia, skin abrasions, and all that. And what we did is we created a network looking at the time similarity to separate the astronauts into different communities. Like I said, we have a ton of data, more than I can go through here. But using this approach, we can see that the astronauts separate into groups that are driven both by the molecular trends of the individual components and we see macroscopically the physiological changes in this set of astronauts that emerge in this group using our machine learning approaches. What I want to do to wrap up is say that AI and machine learning at AWG is very new, but you can see it is pivotal for the future of space exploration and health. And we're collecting this massive information for every astronaut in these missions, and this is going to escalate. This is multimodal data. We have to handle this open data and proper metadata. So we need frameworks and algorithms. And so we have some kind of hefty goals to make the space biology knowledge bases. And I put in here some challenges for everybody to see of things we can do. We have low end we have to handle. We have multimodal big data. We need access to data. Everybody has mentioned that. And maybe we need something, we need knowledge bases and ontologies, something like NASA GPT probably. That would be a worthwhile effort to invest in. Just going to briefly summarize. We've had a bottom-up approach starting from individual assessments of astronauts. We detected individual changes and signatures and change points in real astronaut data using their time series. And we're trying to extend these to non-invasive measurements and using saliva omics. And we all need to help with the development of new AI and machine learning methods. And our goal is to have this AI ML-enabled individualized space medicine. And with that, I want to thank everybody in my lab and our collaborators and everybody leading the AWG groups here. I think this is great work that's been done. I thank Trish for our funding, and questions are welcome. And also I want to say sponsors are welcome for this work. Thanks, everyone.
Thank you so much, George. We have time for one question, and then we do have to move on. So for your time series methods, is it important to incorporate longitudinal data from their time living on Earth? And if so, how far back before a mission is recommended to measure?
It's definitely important to have the data to establish the baseline, especially if you're looking at on the effects of spaceflight itself. I mean, we're talking about tremendous gravitational effective Gs there. So typically the astronauts are going to prepare for going to space. So you want to at least capture their state before preparation, during preparation, and after. So I would say at least 6 months is what we would be looking at.
Wonderful. Thank you so much. We do have to move on, but thank you for your time and for that presentation. So our next speaker is Val Fajardo from Brock University, and he will be representing the Ames Life Sciences Data Archive AWG. Val, go ahead and take it away.
Can you hear me okay?
We can hear you.
Okay, thanks. Thanks for the introduction, and thanks for the invitation. Really, this has been a very interesting symposium so far, and I'm happy to share some of our results with the muscle samples that we've obtained from the Rodent Research 1, Rodent Research 9, and Bion M1 missions. I admittedly think that my title is pretty clever, but maybe it's not. I'm going to try to move. It looks like my computer's freezing. Is this moving okay or no?
Are you advancing? Oh, there we go.
Did you get it here now? Oh,
yeah.
Okay, it looks like it's lagging. Sorry, I have to go back one. I'm sorry. Yeah, it looks like it's lagging. I apologize for the technical difficulties. Just give me 1 second, please. What if I do this? We still see this okay?
Yes.
Okay. And if I transition, we're okay?
Yes.
Okay, thank you. So our lab goal here at Brock University in Ontario, Canada, is to improve muscle health, to improve whole body health in conditions of aging, diseases like Duchenne muscular dystrophy, obesity, and spaceflight. So really what we're trying to do is make our muscles bigger and stronger to attenuate what we know happens with spaceflight, which is muscle atrophy and muscle weakness. And we currently have two strategies that our lab is focusing on right now. And the first one being altering circuit function. And I'm going to spend a lot of time talking about this today before rounding out my talk and talking about the second strategy, which is inhibiting this enzyme called glycogen synthase kinase 3, or GSK3 for short. So the circuit pumps, that stands for the sarcoendoplasmic reticulum calcium ATPase. And they catalyze the active transport of calcium into the sarcoplasmic reticulum, which is this calcium storage unit inside all of our muscle cells. And to do this, the circuit pumps found here embedded in the sarcoplasmic reticulum, or the membrane of the sarcoplasmic reticulum, they use the energy harness from ATP hydrolysis to push calcium uphill. So if you can imagine pushing anything uphill, that's going to require energy, and that energy comes from ATP hydrolysis. Now, this is important for muscle relaxation, but also ensuring a sufficient calcium load in the SR for the next subsequent muscle contraction. So this lightning bolt here, that signifies an action potential. What that's going to do to our muscle cells, that's going to change the membrane voltage in the membranes, and that's going to be sensed by the dihydropyridine receptors shown here in green. And that's going to relay that change in voltage to the ryanodine receptors, effectively activating them. And when they're active, that's going to allow for calcium to be released out into the cytosol, and then calcium will bind to the myofilaments, eliciting muscle contraction. To elicit muscle relaxation, that calcium activating signal needs to be mopped up, needs to be brought back into the SR. Again, that's done by the circuit pumps, which we study quite heavily in the lab. Now, this is all good when it works well, but when we get in conditions of circuit dysfunction, what can happen, you can imagine, if we can't bring calcium into the SR, then what happens is we'll get into this state called calcium overload, where we have elevations or chronic elevations in intracellular calcium. Now, chronically, these elevations in intracellular calcium can lead to a lot of negative consequences. And I've got a list here. It's not exhaustive. It doesn't include everything, but one being calpain-mediated protein degradation, the second being ubiquitin-mediated protein degradation. So two pathways involved with proteolysis, mitochondrial dysfunction and oxidative stress, and also calcium overload can be a signal for apoptosis. And all of these items here, they'll converge and contribute to muscle atrophy and weakness. And so the question that we had originally to obtain our NASA samples from RR1 and RR9 missions was, can circuit dysfunction contribute to spaceflight-induced muscle atrophy and weakness? In other words, what happens to circa after spaceflight? And leading the way on this is a talented PhD student in my lab, Jessica Braun. And she published this paper 2 years ago now, characterizing circuit function in murine skeletal muscles after 35 to 37 days of space flight. And I've shown here also a podcast that she was featured in. She did a really fantastic job explaining her results that were published in this paper, probably a better job than I'm about to do in the next slide. But anyway, we have here on the top left, I can orient you here. This is male mice, 35 days in space, part of the RR9 mission. And then on the bottom left, we have 37 days in space with female mice, part of the RR1 mission. And what I'm showing here are calcium uptake curves. So we assess the ability of the circuit pumps to bring calcium into the SR. And we actually just published a protocol paper. There's the PubMed ID if you're interested in learning how we perform this calcium uptake assay. But in brief, what we do is we provide the muscle samples with a calcium dye called Indo-1. It's a calcium fluorophore, so we can track cytosol calcium levels. And then we provide them with a bolus of ATP. And when we give them that ATP, the circuit pumps are going to take that ATP. They're going to use it as energy. And they're going to start to bring that calcium from the cytosol into the SR. So effectively, you'll see a reduction in the cytosol calcium levels. And we see this quite nicely in both the GC and VIV controls, but what you don't see, what I want to point your attention to are the red lines on both curves. That's the flight group there. You could see that calcium uptake or circummediated calcium uptake is quite impaired in both male and female mice. And by the way, this is in the soleus muscle, a slow twitch postural muscle. And when we look at the area under the curve, which is basically looking at the total amount of calcium taken up over time, you can see that it's significantly elevated in the flight group compared with both GC and VIV controls. We collapsed them in the RR1 mission just to increase our statistical power. So again, circuit function is impaired, and we attribute this to an increase in oxidative stress. Now, these are markers of oxidative stress that we assess via Western blotting, looking at protein tyrosine nitration and protein nitrosylation. So our working model is with spaceflight, this is about 30 days of spaceflight, there's an increase in reactive oxygen and nitrogen species, or these free radicals that can damage the circuit pumps. Now, this damage to the circuit pumps will impair their ability to bring calcium in, effectively raising intracellular calcium, leading to this calcium overload, which unfortunately can contribute to mitochondrial dysfunction, leading to this vicious cycle of accelerated oxidative species production. What's interesting, though, is that Jessica also looked at a fast muscle type. The data I just showed you in the slide previous to this was, again, from the soleus, which is a slow twitch muscle. And just to recall, circuit function was impaired there due to an increase in oxidative stress. At least that's what we think. In the tibialis anterior fast twitch muscle, we found that circuit function was actually enhanced. So if you can look at the curve here in panel A, you can see in the flight group shown here in the red line, if you provide it with a bolus of ATP, they bring the calcium levels down much sooner, and they have a significantly lower area under the curve. Now, we attribute this to the fact that there was a slow to fast fiber type shift. The soleus muscle also experiences a slow to fast fiber type shift, and I'll ask you to pocket that because I'm going to bring that back when I talk about GSK3. But the difference is we didn't find any signs of oxidative stress in the tibialis anterior. So anyway, we left it at this. We published this 2 years ago. We uploaded this data set to the Open Science Data Repository. There's the link there below. And then a few months later, Ryan Scott contacted me and said that there were a few researchers that were interested in collaborating with us with our uploaded data set, and that being Lauren and Sylvain. And I'm not going to try to explain what they've done. I'll just read what they've done here. They used a machine learning approach to predict novel molecular drivers behind our uploaded calcium uptake data. And they took this integrated omics, so proteomics, transcriptomics, and epigenomics approach to figure out what else could be contributing to the differences in calcium uptake data between the soleus and the tibialis anterior. And they really did come up with a few hits, but one of the targets that caught our attention, and to be honest, I've never heard of this until this collaboration. So one of the benefits of this collaboration, this protein called acylphosphatase 1 or ACYP1, which they found was downregulated with flight in both the soleus and the tibialis anterior. And the kicker here is this ACYP1 can enhance circa in slow muscles, like the soleus, and inhibit circa in fast muscles, like the tibialis anterior. So this difference in ACYP1 in terms of their function can potentially contribute to the differences I just showed you with the soleus versus the tibialis anterior. So really neat that this happened through this collaboration, all because we just uploaded our data. And I'm happy to say that Jessica and I are part of this manuscript that will be submitted for publication in the coming weeks. So thank you, Lauren and Sylvain, for that. So just to finish off here, I'm going to talk quickly about the second strategy of our lab, which is to inhibit GSK3, again, with the overall goal of improving muscle health, to improve whole body health in conditions of aging, disease, and space flight. So GSK3 stands for glycogen synthase kinase 3, which is a serine-threonine kinase, and it phosphorylates and inactivates glycogen synthase. So it was first identified for its role in regulating glycogen synthase in muscle. So it does have a role in regulating glucose levels and insulin sensitivity as well. Now it has over 100 cellular substrates, but admittingly, that's probably modest. It's probably closer to 500 cellular substrates. But anyway, why we're interested in GSK3 is because it's a known negative regulator of muscle size and strength. So if we look over to here to the right, we find that when we knock down GSK3 specifically in muscle, these are litter mates matched in weight here, we see obvious increases in soleus muscle size. And what's impressive about this is this knockdown is partial. We've only knocked down GSK3 by about 40 to 50%. So with this in mind, our question with the NASA samples was, well, is GSK3 then a viable countermeasure target against the spaceflight-induced muscle atrophy and weakness? In other words, can GSK3 overactivation contribute to the muscle atrophy and weakness that we see in space? But we know that no one's really characterized what happens to GSK3 after spaceflight, and that's what we wanted to do. And leading the way here is Ryan Baranowski, who just finished his master's degree in my lab, but he's sticking around as a lab technician, thankfully for me. And what he found was actually quite surprising. He found that GSK3 was significantly downregulated with spaceflight, and we assessed this through Western blotting, but because of the 3 different missions that we had, we kind of took this meta-analytical approach where we collapsed the ground control and Viv controls because there's no difference between the two. And so what I'm showing you here is a combined fold change. And what we can see is that there's a 30% reduction on average across all missions in GSK3 content. And that begs the question, well, if it's downregulated, then is it really contributing to muscle atrophy? But alternatively, maybe this downregulation serves as an adaptive or physiological response aimed at mitigating the muscle atrophy and weakness that we see with spaceflight. But more importantly, the question is, well, what is causing this downregulation? And Ryan just finished up some really neat immunofluorescent experiments showing that in normal conditions, this is just in soleus muscles from loaded wild-type healthy mice, you could see that GSK3 here is highly expressed in these green type 2A fibers, okay? And I asked you to pocket that slow to fast fiber type shift, and I'll bring it back here. With spaceflight, we do see a slow to fast fiber type shift, okay? So the muscles are comprised generally of slow oxidative muscles and fast glycolytic fibers. And with spaceflight, what we see in the soleus is a significant reduction in type 1 and a significant reduction in type 2A fibers, and then an increase in the glycolytic 2X and 2B fibers. So if we see a reduction in the type 2A fibers, thank you very much, then it makes sense, because the GSK3 is mostly expressed here, it makes sense that there's a significant reduction in GSK3 content. But then the next question is, well, what if we inhibit GSK3 earlier on and before this fiber type shift occurs? Because keep in mind, these samples, they were obtained 30 days after space flight. So our question is, well, what happens if you inhibit GSK3 earlier on? Okay, and so I'll show you some of the GSK3 knockdown experiments and hind limb suspension data that we have, and thanks to Brianna Hockey for generating these mice, these GSK3 muscle-specific knockdown model. We couldn't have these results without her. So anyway, what Ryan has shown is that when you knock down GSK3, and even under hind limb suspension, the soleus muscles are still bigger. There's a greater proportion of larger fibers. There's a rightward shift in the cross-sectional area distribution curve. And these arrows, they're pointing at centrally located nuclei. So we see an increased presence of central nuclei, which to us tells us there's increased myogenesis, which is really important. And this is corroborated with a significant increase in PAX7 and myogenin, which are markers of myogenesis as well. And in panel G here, I'll just finish off really quick. This is a force frequency curve looking at specific force production across submaximal and maximal frequencies. And the knockdown shown here in red, you can see that they are producing more force compared with their wild type. I'll call them wild type controls. And I'm not showing you the data, but when we look at normalizing this to mobile controls, it's actually a greater preservation of force, where the wild types had about a 50% reduction in force versus the 27% reduction in force when you knock down GSK3. So anyway, I'll leave it there, and I'll take questions, but I'll say thank you to my lab, especially Jessica Braun right over here, Ryan Baranowski, Brianna Hockey. They make up the space team of my lab, and so thank you to them. Thank you to the collaborators. I'm probably missing quite a bit here, and I apologize for that. Thanks to the funding and thanks to NASA, and of course, thanks to the organizers of this conference. And sorry for the technical difficulties before.
No worries. Thank you so much, Val. We do have a question for you here. GSK3 is the downstream of insulin. Did you look at insulin or insulin receptor expressions?
We have not looked at the insulin receptor expression, but very good question, and we will be looking at that. And I do want to say that Brianna Hockey is leading a project with high-limb suspension with our Canadian Space Agency funded work, looking at whether or not inhibiting GSK3 with lithium can attenuate muscle atrophy and weakness in the high-limb suspension model. And we will be looking, hopefully I can convince Brianna, I think she's on the call, to look at insulin sensitivity.
Wonderful. Thank you so much, Val. Absolute pleasure to have you here. And we do have to move on to the next speaker.
Thank you.
So the last speaker in this particular section is Laura Fackrell. She is from NASA Jet Propulsion Lab, and she will be representing the microbes AWG. Take it away, Laura.
Can you hear me all right?
Sure can.
Okay, perfect. Okay, let me share the screen. Okay. Good afternoon. I am Dr. Laura Fackrell, and I am working with the Biotechnology and Planetary Protection Group at the Jet Propulsion Lab as a postdoc. And today I'm going to be talking about a nanopore sequencing and DNA extraction project that we're working on with some lunar regolith simulant-based materials. Before getting into the actual talk, I just want to acknowledge my fellow authors and committee and all the support by funding and also just general support that helps to that's helped to kind of start this project off. I'm going to do just a really brief context to kind of give an idea of where these samples are coming from and what that has to do with the microbiology of the situation. And then move into the main point of the talks, which are kind of like the challenges that we're trying to address with that and some of the adaptations and approaches and questions we're trying to answer in our approach, some very, very early results and kind of what we're looking at for the next steps. So a really brief challenge, a really brief context for this to kind of show where these samples are coming from. And they're coming from our project where it's actually a plant project where we're looking at certain aspects of growing plants in the space, not just if they will grow in general, but also just like looking at it from a system kind of perspective to see different things about what you would do with plant waste, how you might recycle that, different things about recovering nutrients and different aspects related to those kind of issues. And how this relates to the microbiology of that problem kind of goes back to a little schematic that I like to show for why we're approaching it in this way. So if you look at kind of like an earth soil for something as an example of one way that you could grow a plant is in earth soil, something that they've evolved to grow in. And you have this material that has that's made of mineral and organic matter, and it has air and water, a ratio that's beneficial. But a really important part of that mixture is the microbiome that's located in the material. Even though it makes up a small weight percent, it makes up a huge aspect as far as the chemistry and how it affects it and how it drives a lot of the chemical processes that happen. And there's a really important synergy in between the plants and the microbes as they interact in this environment. But as we switch to the moon, you suddenly change that entire picture where your mineral material is still there, but whether or not it's useful for the particular application that you desire is another question. And you don't necessarily have an atmosphere anymore. So rather than air between the grains, there's maybe just space or other volatiles between the grains, and the water maybe is there, but it's frozen or another kind of useful, less useful raw state. And then you've kind of eliminated your microbes and your organic material that are part of that initial environment. And so it kind of completely changes the situation. And then, of course, with processing and using the utilization of those in situ resources, hopefully you can process those materials so that your mineral and product is now kind of at a state that's useful. You put the water back in a state that's useful, and it's in an atmosphere that's ambient. And so it's going to, again, kind of represent that part of the picture. But there is a pretty big gap that remains with how we might proactively influence the microbiology. As you bring it into the environment, certainly it's exposed to the microbes, but how do you proactively try to influence that microbial community that is existing in that environment, and in particular in association with those plants, so that it's actually helpful? And then what can you do to help kind of mitigate? You have a lot of organic matter that's coming from waste, but how do you get it back into the actual system and make it useful? So that's kind of what the project is looking at, is trying to kind of complete that picture in that kind of sense. And so that's where the samples are coming from, and why we're kind of looking at that aspect. And then that ties back into the microbiome in the sense that that's one of the aspects that we really want to dive into, is how do we proactively influence that microbiome so that it can be one that would help increase plant health, and or at least support plant health, and help to make a healthy plant microbiome that the plant microbiome can be healthy, that's associated with that plant. So that's part of the project, and so we want to look at the microbiome. So that's why this is the microbiology side of it. And that kind of brings us to the point of this talk, which is there's a big challenge with that, in that these materials, although they are soil-like, are not necessarily soil. And so that represents a few challenges in how we might want to do that work. And so that's something that we thought might be useful to look at in general, as these simulants and as different simulant materials are being used in these applications. And as far as those simulant materials carry forward to actual materials, actual regolith and actual processes that might involve those, it's useful to know how such protocols and things that we use all the time for all these different things, and validate their application to these new materials. And so part of the problem, so looking at the different materials, we have the actual simulants. They are, although lunar regolith itself is not expected to contain any kind of microbiome, microcommunity, or anything living, the actual simulants that represent them certainly do, as they're not created in sterile environments. And so those simulants have their own endogenous community that comes from just how they're processed and created. And that tends to be pretty low biomass, so it's hard to extract in the first place. Now that could be sterilized into, depending on how much material you're trying to do at once, and what type of sterilization approach you want to use. And so that can be a good way to just kind of eliminate that, and maybe cut out that factor. However, how effective is that sterilization? We need to be able to test the effectiveness of the sterilization. And there's also a lot of situations where the sterilization process actually affects a characteristic that's important to the question you're trying to answer. And so you might not want to actually sterilize, but you still need to be maybe aware of the microbiome that's already there, so that you can take account for that into your results. So there's a lot of issues there that kind of want to help out. And it's this, lunar simulants aren't necessarily super complicated, in that it will probably readily carry over to, depending on which simulant you're using, but it gets a lot more complicated as you kind of dive into other simulants where there are a lot of salts or different things that could really affect how these extraction kits work. And so that's kind of a part of the challenge, is how do we address that? And then another part of this soil-like materials, that not just the simulants themselves, but also these materials that we create from the simulants. So all these different materials that come from that would, for our experience, we want to track, but also if similar materials were ever created in an actual situation, you also want to track that. And so we want to make sure that these things are actually, they work with these materials, which, and so just trying to look at the efficacy of that, because they're, even though they have soil-like properties, they also have things about them that are very different from how an actual soil would behave, or even an actual, or a typical compost that you would have in an earth setting would behave. So it's just making sure that these still carry over. And we have a variety of different materials within our own project and within the community at large. All the different simulants that are used and all the different things that are applied can be very different from each other, chemistry wise, and are intended for different applications. And so making sure that that carries over and that there's a connection there. And also not wanting to overcomplicate the situation, because you do want to be able to apply it eventually to situations where it's maybe a little bit trickier to do lots of different chemistry with lots of different pieces. And so you need something that's more readily automatable. So that kind of brings us to the project that we're working on right now, which is kind of just like a, just kind of verifying the protocol and adapting the protocol sort of project. And there's 3 questions that we're really wanting to approach in this project. And that's that what adaptations are even needed in the protocol, if any, or if the protocol as is works well with these materials, and that's great. But what do we need to adapt and how is that working? We're also working on the being able to verify the sterilization techniques, both on the side of does it actually sterilize? But the bigger question there is, what does that sterilization do? And to what extent can you, depending on what your questions are and what the study is, how important is that into maybe that you might have to consider skipping sterilization if it affects characteristics that are really vital to your questions that you're trying to answer with your approach, then, which leads to the third question is like, well, what even lives in regolith simulants that are in multiple labs in multiple places and have been created from multiple different sources and have been exposed to multiple different environments? What are even in these simulants? And is there a way to kind of like create something that could track that in a more accessible way that could be a resource for the community? So that's a part of that question. And so we're at the very, very beginning of this project. So what I'm talking about today is we've literally taken a few samples and run them at one time. So we don't, so it's not at a final stage yet by any means. So we're kind of just starting. But what we have been able to kind of approach so far is just using what's available out there and just starting and seeing how viable it is. And so that's included a DNA extraction kit with QIAGEN, the DNA paraprocell kit, and then also making sure that we've kind of use those protocols pretty much as they are, but with slight adaptations for things that we already know. For example, for the samples that we know are low biomass, making sure that we do things that can help increase the biomass at the end, and then testing that the quality of that DNA. And for samples that were very low, we had a few samples that were barely readable by qubits, but for other samples it was pretty rote, and so we were able to do nano per dropper and look at both the quantity and the quality of the DNA. And also looking at library, we did, for this particular round of samples, we did a rapid barcoding kit using the Kit9 chemistry, including a cleaning step to kind of help with some of the impurities that we saw in some of the DNA, and use the MinION to kind of do the sequencing side of things. And that put everything together, and then just did a very, very quick crack-in kind of metagenomics just to see what was even our starting point. And so that kind of leads to this side of things. We're these very early results. So this is just like a very rough run, nothing finalized yet, but it kind of gives us an early hint as to where we're going and things that we might be able to do next. So there's some really cool initial things that we're seeing, both in the actual simulants themselves and like what might be interesting to look at and to dive into with that, and some things that we're very excited for. And also just like looking at the changes that are happening to our materials that we're actually creating for the main project, and the changes that are happening at different stages of that processing, and what we can see for what's happening to the soil microbiome, or the soilless microbiome for a soilless media and that kind of case. But it's really interesting. So we're really excited about what's to come. And so we have those early hints at one, what lives in regolith, and then two, what are the dynamics of these communities that live in this material that's developing as the project moves forward. So kind of next steps is we definitely some things that though we have some interesting results so far, a lot of things to change about the protocols, in a sense of like making sure we have viable data and everything. So we're working on those next steps and some of the ideas that we have. And we also want to then, once we kind of more refine that, get to the point where we can do like test different mock communities and seed them with that, and look at the sterilization techniques in different aspects. So there's a lot of things that are coming with that, but that's kind of the very beginning of what we're starting with that kind of project. So that's all we have to say, and take any questions.
Awesome. Thank you so much, Laura. So we do have some questions for you here. Let's see if we can get to them super quick, because we are a little over time. Do you think that these soil-like substances might have different water wicking properties, and does particle size matter?
Yes, yes. So yeah, I would say there's definitely different wicking properties, and that's going to be really important for how they behave in like different gravities. It depends on what simulant you use, and it depends on what your organic matter that you're using is. So we're using the same thing and the same thing. So it's been pretty consistent, and so it's a pretty homogeneous material at the end, just because we're using like pretty simple starting points, but it gets more complicated once you have a greater variety of things to include. And then I forgot the other half of the question, but
particle size, does it matter?
Oh, I would say yes, it does definitely matter, because that affects the balance of air and water in the system, and then how things swap onto other things. And so there's a lot of things that the particle size will affect. So definitely an important aspect.
Great. Last question here is, do soil simulants have accession or batch numbers? So OSDR could account for those differences.
I think it depends on who's making it and how thoroughly they track that. And that's definitely something, so that would be an important aspect of like, maybe that's something to consider in how we use these things. And that would be kind of, how do you track that? And so so this is like a lot of things to think about if you want to actually track what's in a simulant and like what's living in these simulants. So yeah.
Wonderful. Thank you so much, Laura.
Yeah.
We have concluded this section of the event. Right now, we will be taking a 10-minute break, and we'll reconvene at 12:50 p.m. Pacific time or 3:50 p.m. Eastern time. So we will see you all in about 10 minutes.
Recording stopped.
next section of speakers will be the AWG Science Lightning Talks. And then this section, the speakers have 5 minutes to talk. Speakers, you will hear 1 ding of a bell, and that will be your 30-second warning, and 3 dings of a bell means that your time is up. For the audience, I'm going to kindly ask that you we will wait until the end of this section in order to do a Q&A for all speakers. So I'll kindly ask that when you put your questions in Conference.io, please add the name of the speaker so I know to whom to address the question. So with that, I would like to introduce our first speaker in this section, and that is Andrea Camara from the University of Oslo, and he will be representing the animal AWG. And Andrea, take it away.
Hi everyone, so can you see my screen?
Yes.
Yes, perfect. So it is a great honor for me to be here today, and I will present you our project on spaceflight and frailty. As you can see, we are many people in this group, so there was no space in the slide for taking everyone's picture, but of course I will present on everyone's behalf. So first of all, I'll start introduce you what frailty is. This is a quite novel topic in the clinic, and it is this term is mostly used in geriatric medicine. So frailty is an age-associated clinical syndrome that is characterized by a decrease of biological reserve and, most importantly, of the resistance to stressors, such as an acute disease or a surgery, and this due to a functional decline of several physiological systems, such as the immune system, the musculoskeletal one, the central nervous system, and so on. And this condition results in an enhanced risk of disability, hospitalization, and death. You have to think that frailty is considered the most important risk factor for the development of non-catastrophic disability in the elderly. And to date, the assessment of this condition is based mostly on physical parameters such as gait speed, weight loss, grip strength, and declining cognitive function and the presence of multiple diseases. And despite this syndrome being usually regarded as a condition present mostly in the elderly, clinical studies demonstrated that chronological age is not the only determinant of this condition. Instead, the pathophysiology of frailty seems to be strongly connected to the biology of aging. And biologically speaking, for instance, a 70-year-old patient can potentially be more aged than an 80 years old or 80 years old one. And as you can see on this slide, there are many processes that are involved in the development of aging, such as DNA damage, mitochondrial dysfunction, inflammation, oxidative stress, and metabolic imbalance. So we decided to investigate whether the attrition caused by the exposure to the space environment could trigger these biological processes. And we hypothesized that these alterations could all together manifest as a frailty-like phenotype in us. And to do so, we focused on the muscle tissue, peripheral blood mononuclear cell, and cell-free epigenome data. And the muscle tissue is so far one of the most investigated tissue in frailty studies, and that this is why we gave a privileged focus to sarcopenia, which is a reduction in muscle mass and muscle mass and function and quality. So this is an overview of our project. We work with, as I was saying, with muscle tissue biopsies mostly from NASA OSD data. We also have the JAXA astronaut study cell-free epigenome and the peripheral blood mononuclear cell inspiration 4 data. With these 3 datasets, we investigated whether a frailty biomarker list that we derived from the literature had this biomarker differentially expressed pre-flight, during flight, and after re-entry. And we also performed gene set enrichment analysis and metabolic flux simulation based on transcriptomic data to find signatures related to aging, such as biological pathways. And also, thanks to the contribution of Professor Caseido's group, we curated a short gene list of predictors of sarcopenia that we obtained by analyzing a population of age-matched individuals with and without sarcopenia, and we assessed their expression in our rodent open science datasets mentioned before. We detected that multiple frailty biomarkers are differentially expressed during spaceflight, and also novel sarcopenia-related biomarkers that we identified, some of them are differentially expressed. So we postulated, we hypothesized that the space environment can somehow influence our development, can cause the development of a frailty-like condition in space. And since even though our results are preliminary, we demonstrated the feasibility of this kind of study, and also we showed the need for further investigation in the field. We submitted a paper that now is under review on Nature Communication Medicine. And as a future perspective, we want to integrate our transcriptomic pipeline to physical phenotypes to better detect frailty in space and develop a minimal panel of biomarkers to investigate. So thanks everyone for your attention. And if you want to join us, just email me or Afshin.
Wonderful. Thank you, Andrea. Our next speaker in this series is Jonas Elsborg from ABZU, and he will be representing the multi-omics AWG. Go ahead and take it away.
Yeah, thanks, Lavarka. Not much time here, so let's get going. Yeah, I'm Jonas. I'm come from the Danish startup company called ABZU, where I work as a machine learning researcher. And this talk is titled Elucidating Dermatological Changes in Spaceflight with Explainable AI. So this sort of reveals that the approach we take here in ABZU is based on the fact that we try to not only predict based on the data that we have, but we also want to try and explain the models that we find. So typical paradigm in AI is that you have this big black box, a tree model or a neural network with many different parameters. You shove in your data and then you get out some prediction, but it's typically quite hard to understand exactly what is going on. So instead in ABZU, we take the approach where we try to implicitly and inherently encode transparency into our models, making them more creative and also simpler, typically, while retaining some robustness. And this also makes us able to interpret them and get out some scientific knowledge. And this is basically why I'm here today, because we took this approach also in a paper that is under review right now at Communications Medicine, which is co-first authored by me and Henry Cope, who I believe is also present today. And as you can see, many familiar faces. We investigated basically what changes occur in skin when you go to space based on transcriptomic data sets from mice. We also included some human data, but time constraints forced me to focus mostly on what we did with the mice in this case. So why even talk about this skin problem? Well, astronauts tend to have skin problems when they go to space. So it would be quite nice for them if we figured out a way to handle that. And on the other hand, since, well, skin is our largest organ, it's like our outer layer, so to speak, protecting us against any external pathogens. So typically any pathology you see in the body, a lot of them will also be like have reverberations in the skin, so to speak. We all know skin cancer, for example. So there's a good example there. But on the other hand, from a more pragmatic perspective, skin is easily accessed, it's easy to probe, you can take a sample quite non-invasively, doesn't really hurt. And well, if we knew how to solve these skin issues, we probably wouldn't be having them when we go to space. So basically, this is understudied. Our data consists of normalized gene expressions from bulk RNA-seq from the MHU-2 JAXA mission and the Rodent Research 5 and 7 missions. So basically, you have thousands of genes, their expression, and then spaceflight status, whether the mouse has gone to space or not. In this paper, we took a kind of a different approach, did some method development specifically for it, because we noticed that a lot of the genes tend to cluster together compared to their sort of functional annotation in gene enrichment. So what we wanted to do was instead to characterize a gene based on what friends it have, so to speak. This basically means that we take a gene and then we ask this proprietary algorithm we have to come up with or nominate another gene that, given that you have information about this first gene, what other gene would you most like to know something about as well to better predict the outcome? And we do this in a quite robust way such that we are quite sure that this specific interaction, this measure that we developed and is detailed in the paper, is solely indicative of the interaction between the genes. So basically what genes pair well together. And when we draw this out, it's quite obvious that, well, also from this visualization where every single edge corresponds to a mathematical model between the two that go towards the output, that some genes are much more central than others. Some have an insane amount of partners, basically a measure that we have of whether it's a good partner or not, compared to others. And this specific gene, BERK5, also known as survivin, is the most frequent one, but it is also the one that has the strongest synergy effect out of the thousands of models that went into creating this picture of the data. And I should say, by the way, that this data is, of course, a reduced set, which Henry was had a genius insight into how to do, which I don't cover here. So the thing is that, well, this model that we find with BERK5 is basically, well, BERK5 is an apoptosis inhibitor, and it pairs well with these proliferation genes. And the model that comes out of this is basically a gene where we can see that the wild type round mice are in an apoptosis proliferation balance. And you can basically swap the partner gene for any of these proliferation and tumor progression genes. And this leads us to the conclusion that, well, space looks like it's a loss of cell homeostasis, at least in the skin. And these mice astronauts did not have this balance while the ground mice did. Thanks to Afshin and Henry for this paper. We did a final stretch here, and thanks to Sylvain and Lauren for always inviting us to this. And on behalf of the whole company, thanks for inviting us today.
Wonderful, thank you, Jonas. Our next speaker is Balaga Aolanreju from Ohio University, and he will be representing the plant AWG. Balaga, go ahead.
Thank you so much. You can see my screen, right?
Sure can.
All right. So I'm Balaga from Dr. Sarah White Lab in Ohio University, and I'll be talking about our project, which is delineating the proteomic response of Arabidopsis to the spaceflight environment in break hardware. The most comprehensive, I don't know, is it rotating? Is it scrolling? Oh, I think
no, we're not seeing you advancing the slide.
It's glitching. Okay, all right. So the first and comprehensive transcriptomic and proteomic study done in spaceflight on Arabidopsis by Cruz et al. In 2020 indicated low correlation between differentially expressed transcript and differentially abundant protein, which is an indication of the presence of post-transcriptional machinery, and also pointing to the importance of proteomic study, that proteomic study can give insight which could not be seen from transcriptomic study. However, due to the inherent challenge of little mass, tissue mass available for proteomic studies in spaceflight experiment, only few proteomic studies on Arabidopsis has been conducted so far. The two proteomic study which are indicated with arrow were conducted in our lab by Cruz et al. In brick PDFU, where the plants are where they grew in the absence of light and in the absence of gravitational cue, and the other one conducted by Olanrewajo et al. Had plants growing in the brick LED hardware where they grew in the presence of light while in space. Also, it is important to note that there was a difference also in the seed line. The experiment conducted, the proteomic experiment conducted by Fell et al. In 2015 were on mutant, whereas the other three data sets were on wild type Columbia 0. So we did a meta-analysis of the protein expression in the four hardwares, and the intersect had three proteins common to the all proteomic experiment. We have the microtubule destabilizing protein, we have a protein involved in defense response, and we have protein involved in mRNA binding. And also excluding the ABRS, the experiment conducted in the ABRS being a mutant seed line, and comparing the three other hardwares which were the wild type. At the intercept lies 8 proteins, and these are the 8 proteins, and a protein-protein network interaction analysis of these 8 protein reveal that central to this protein, to the function of this protein, is a regulation of microtubule polymerization and depolymerization, which is indicative of the fact that microtubule polymerization and depolymerization is a major adaptation of plant to the spaceflight environment. Now, excluding limiting our experiment to the 2 conducted in our lab, where we have one conducted in the brick LED, which had light, and then the brick PDFU, where there was no light. Intersecting both of them, at the intersect of these 2 experiments, definitely will be proteins that are abundant due to the spaceflight environmental stress condition, microgravity, and ionizing radiation. Here we have protein abundant due to photomorphogenesis, cross-interacting protein, cross-interaction between light and the spaceflight environment, and elusive protein due to advanced technology used during the proteomic analysis of the brick LED compared to the brick PDFU. And here we have protein that are involved in scotomorphogenesis, that is growth in dark, and cross-interacting protein between darkness and the spaceflight environment. Now, bringing this intercept, which I call brick intercept, and comparing it to another experiment conducted in the European Modular Cultivation System aboard ISS, where we have 1G in space. That means where we remove the condition of microgravity. So at the intercept of both of them, we definitely be proteins that are abundant due to radiation, ionizing radiation in spaceflight, and due to fluid gas mechanism alteration, which is not really important, when dealing with plant as at that. And here we have proteins that are expressed majorly due to microgravity, and here proteins that are expressed majorly due to the European Modular Cultivation System hardware and cross-interaction between the hardware and spaceflight. Now, conducting a protein-protein interaction on the 14 proteins that are expressed at the intercept, we have we see that majority of this protein are involved in ribosomal activity. And that is also taking note of the fact that some of them are involved in regulation of microtubule depolymerization. Now, from this, it's obvious that the target, one of the major targets of spaceflight ionizing radiation for Arabidopsis is the ribosomal activity. And so currently we comparing the transcriptomic data of the brick PDFU and the brick LED experiment, and also we will be proceeding to wet lab determination of the specific roles of this intersecting protein. And of course, we open to collaboration on proteomic meta-analysis. And so I acknowledge Dr. Sarah White, Dr. Colin Cruz for their inputs during this data analysis. And I also appreciate the NASA AWG Plant Omics group. Thank you.
Thank you so much, Balaga. Our next speaker in this section is Aidan Lewis from Colorado State University, and he will be representing the Artificial Intelligence Machine Learning AWG. Take it away, Aidan.
Awesome, thank you all. Appreciate you all for coming to my talk today, Personalized Medicine for Martians. I, like many of you, really want to go to Mars, but space is really, really stressful for our body. Oh no, it's not advancing. Oh, there it goes. Oh yeah, it's lightning tigers, lightning bolt. All right, here's an overview of what we're going through today. Just going to do a little background on what space for AI medicine is about, the approaches in my research, some preliminary data, and then the future directions of my work. So let's get started with the background. Here's kind of the overarching goal of using AI for space health. Can we take a person here on Earth, get some data from them, feed it to some AI ML, and then be able to predict how they're going to respond to long duration space missions and living on Mars? Obviously, this is a little bit of a problem because there's very limited data for astronauts in particular, and analogs are analogs at the end of the day. So we're working right now on being able to make these models count right now with some different data and then build on top of them. And here's just that in a picture view. So right now I'm working with a rhesus monkey data set that I'll talk about in a second, using these health analogs to build some models that we can then incorporate further data, such as human data, to then further refine them and hone them to make really powerful predictions for spaceflight and living on Mars. So what's the approach? Here's that monkey data set I'm talking about. This is about 275 monkeys housed at Wake Forest University. It is a public data set and they really want to share it with the world, and they're going to come out with a package of papers delineating that. But what's unique about these monkeys is that some of them have been irradiated at different points in their lives, and they have meticulous health records kept, monthly complete blood workups done, CT scans even. So these monkeys have just had the full gamut of health data collected on them. So what I'm doing is I'm using cat boost gradient boosting models to see if we can do things like predict how the monkey's biomarkers change, eventually looking at the central nervous system using some computer vision. How does it change due to radiation? Can we cluster monkeys based on response? And then do things like predict health elements like tumor genesis at the end of the day. And there we go. Some preliminary data is I predicted lymphocyte counts, which is pretty cool. So I chose lymphocyte percentages for these monkeys because it is a biomarker that's responsive to radiation and was one of the only biomarkers that actually showed some differences between the population of monkeys. And so what we're looking at here is on the y-axis, what the model predicted the lymphocyte percentages to be versus what the actual lymphocyte percentages are. On the left is just doing this for one monkey. And then on the right is doing this for the full monkey cohorts. This is 30%. So this was a 70-30 split. This is predicting the 30% here. And I will note that this is off of 10 iterations in learning, and we're getting R squares of 0.9 and above. With 2 to 4 iterations, these are R squares of 0.8. So these are very sensitive models for this type of data, which is really impressive. You can do this with 5 different features. So what we're looking at is about 26 different features. You can do this with just irradiation data. You can do this for less monkeys. So even with 20 monkeys, this is still accurate. So really, really powerful stuff. And the application being a diagnostic and risk mitigation tool for NASA. I'm not going to show this as well, but you can also predict the occurrence of tumor genesis in these monkeys with high accuracy and precision as well. So very, very exciting stuff. Cool, so future directions. What I want to do is start incorporating this data in these models with other NASA data, such as the OSDR data, the standard measures data, and really make these really robust ensembles of models. Also implementing NASA tools such as the causal inference platform CRISP and the impact tool that the XMC XMC folks have developed as well for predicting the likelihood of different elements occurring in space. And so incorporating these powerful models into these other powerful models to make them even more powerful. That's why I like it. This is a very fast talk, so if this is interesting to you, please contact me. I love talking about this. My LinkedIn, but thank you so much to the Bailey Lab here at CSU, Dr. Lauren Saunders, we're getting involved here, Dr. Klein, who's at Wake Forest and who has the monkeys, NASA, everyone who's believed in me, appreciate it. Have a good one, y'all.
Perfect timing, Aiden. Thank you so much. Our next speaker in this category is Matthias Nesset from McGill University, and he will be representing the Ames Life Sciences Data Archive AWG. Go ahead, Matthias.
Okay, thank you, Lavorka. Second as we get this going. All right. Hello, everyone, and welcome to a lightning talk about my project, the Citizen Science Approach for Searching and Curating Literature on the Effects of Spaceflight on Cardiovascular Outcomes in Rodents and Humans. Spaceflight experiments are costly and rare, making the legacy data they acquire important for enriching datasets. Therefore, there's a significant need to find, curate, and incorporate legacy data into AI-ready databases such as NASA Ames Life Science Data Archive, or as I'm sure you all know, ALSDA. Previously, our lab curated legacy data on bone health and space travel using knowledge synthesis approaches, a paper from Fu et al. In 2021. This caught the eye of Ryan Scott, who then contacted my supervisor with the intention of uploading this literature-derived data into open databases. From this paper, we then selected 5 of the 40 articles used, which were NASA funded and already existed within NASA databases for curation into ALSDA. During our data curation and upload process, we ran into several different problems that required some problem solving and iterative efforts. I don't want to spend too much time here, but I just want to outline that this table is just a small fraction of the obstacles and solutions we encountered during this time, with the main one centering around the required configurations at the top. Sorry, I'll just add my pointer with the ones centered around the required configurations and having them available to be uploaded. Here, we have a short process flow diagram of the curation process. A high-end view shows that it's effectively split into a 3-step process with assay configuration, data templates, and data curation, with reviews from various experts denoted by the stars along the way. Finally, a few weeks ago, I had received word that one of the studies that we had curated fully cleared approval and was publicly released. That was very exciting for us. And this can actually be found as, it was previously known as GLDS 471, but now as OSD 471. And here's what part of it looks like. Now, extending this bone outcomes analysis, we know that the spaceflight environment causes significant changes in the structure and function of the cardiovascular system. Going back to what I mentioned earlier about the need to find, curate, and incorporate legacy data into open databases, regarding my project right now, we are currently in the finding stage. Consequently, our overall goal is to find, extract, and analyze all the recorded data for cardiovascular outcomes in spaceflight and simulated microgravity. To do this, we took a multi-step citizen science approach. We started by approaching NASA ALSDA Analysis Working Group investigators, where we held 3 meetings with 25 investigators and developed a list of relevant terms. These terms were then transferred to librarians who executed a search strategy in Medline, CINHL, Embase, and NASA repositories, yielding just under 19,000 articles. In parallel, we decided to recruit students and young professionals from space industry affiliated organizations. At recruitment close, we had approximately 100 individuals join from over 28 countries with a variety of high-profile educational backgrounds. Finally, we offered all participants a virtual training course on the nature and methodologies of the project. Looking at actual screening progression, initially screeners worked at a decently slow rate of 5,000 articles per month. However, my supervisor and I then followed up with the top 20 screeners and gave them more tailored training, which increased their screening rate by 12 times to about 60,000 articles a month. And no, that's not a typo. Finally, these articles were then labeled into relevant categories. This Prisma diagram shows our screening results. Of the approximate 19,000 articles we had started with, only about 3,500 will be going through to full text screening and resultant labeling. Now, touching on the labeling process, the main groups of the labels are shown by the headers: language, population, experimental methods, the simulated experimental methods, the focus of the article, and the study type. Additionally shown are each subgrouping of the articles and labels. Labeling results here show that the majority of the articles are actually in English, done on humans, performed and simulated in actual spaceflight, spread out amongst simulated methods, while most of them focusing on cardiovascular outcomes and being primary studies. As outlined by the title of this project, we are focusing on large subgroups describing outcomes of actual and simulated spaceflights on humans and rodents, while also considering some radiation effects. Specifically with regards to radiation, we realize that there are some aspects that we missed in our initial search, and thus we will continue to be working closely with AWG members to redo this part of the project for more completion. We successfully showed that information science can reduce, informed citizen science can reduce screening time from months to weeks. Additionally, we recommend that investigators screen 10 to 20% of their library to better understand some topics that might require clarification. And finally, we showed that smaller individual work sessions drastically boost screener confidence and productivity. Looking at the future work, we want to do due diligence checks on the screening results by cross-checking the references of the reviews that we included and asking ALSGA experts to spot check articles they know should be included. We will complete the full text screening with the aid of our top 20 screeners. And after this, all of the data extracted will be added into the ALSDA OSDR open database using methods similar to the bone data upload. Thank you for listening, and thank you to all the collaborators who have helped.
Thank you so much, Matthias. Thank you for your time and your presentation. And our last speaker in this section before we get to Q&A is Anna Simpson. She is from NASA's Jet Propulsion Lab and will be representing the microbes AWG. Go ahead, Anna.
All right, can you hear me?
Sure can.
Let me share my screen. Okay, can you see the full slide?
Yes.
Okay, great. So hi, everyone. My name is Anna Simpson. I am at Jet Propulsion Laboratories Planetary Protection Group, and I'm here to present on monitoring of ultra-low biomass JPL spacecraft assembly facility surfaces using novel surface sampling and nanopore sequencing, so more at the pointy end of data collection. Please advance a slide. I will ask you so nicely. There we go. So NASA planetary protection is policy is that space hardware must be certified to a certain level of biological cleanliness. But right now, the standard assay for certifying hardware selects specifically for bacterial endospores and takes 72 hours. So one, you're not catching your non-culturable microbes, and two, that's a really long time to wait with your delicate spacecraft instrument open and unable to move forward with putting your hardware together. So ideally, from both a planetary protection and an engineer's point of view, we would be able to sample, do rapid DNA extraction and sequencing, input the data into a risk assessment model, and within 24 hours or less of sampling, be able to say whether equipment needs to be re-sterilized or whether it can be certified for flight. And if you want to say commercial spacecraft company or spaceflight companies to follow planetary protection standards, whatever you ask them to do, it has to be easy and it has to be quick, or they are not going to do it. However, the gold standard Illumina library prep time and sequencing time is pretty prohibitive for rapid prep. And so nanopore sequencing is like the obvious choice here. It's light, it's portable, it's easy, but it's not designed for ultra low biomass samples. So we're going to need to do a lot of QC to make sure we're getting good quality data. And there is always the ever-present issue when doing ultra low biomass work with background contamination from reagents, which in the case of human environments, sometimes the very same microbes that are in your control reagents are in your samples. So here I'm going to present us trying this out on the JPL spacecraft assembly facility floor, along with a new sampling instrument with much higher efficiency than swab or wipe, and seeing what the kinks are. So I'll quickly take you through the steps. So we took samples from the clean room floor using a newly developed sampling instrument called the SALSA, aka Squeegee Aspirator for Large Sampling Area, which is patent pending and funded by a NASA SBIR grant from AI Biosciences. Its mode of sample collection should be pretty obvious here from this picture. You spray sterile water and collect it in the tube, and this sampling head here that you see is replaced with a new one out of the package, along with a new collection tube each time so that you're maintaining sterility. We sampled 3 locations in the clean room, 2 high traffic areas, so the A shower or AR in this figure, the entrance area of the clean room, ECR, and 1 low traffic area at the far end of the clean room. So we sampled 3 12 by 12 inch adjacent areas in each spot, and also took 3 process controls where we sprayed the same collection water that we were using on the floor into a sterile collection tube and sucked it up with a SALSA instrument. So it was going through all the same processes that the control samples were. And so from there, we proceeded to sample concentration, DNA extraction with metapolyzyme, followed by an automated magnetic bead kit designed for low biomass, library prep with Nanopore's rapid PCR barcoding kit, which involves random amplification with long amp TAC, bead cleanup and concentration with some alterations to use more DNA in the PCR step, and pooling and concentrating the tagged DNA before library prep, and sequencing on an MK1C. And so we ran it for 24 hours just to see how much data we would get. But if we'd run this for 3 hours, we really would have been able to do this by the end of the day. So this is probably the most important bit of results that we got out of this, which is here. So here we see the actual samples on the left side of the graph. So ECR, FCR, AR, and then we have the controls on the right side of these graphs. And so on the top left, we have the qubit of the original sample. Graph B, we have 16 SQ-PCR results. Graph C, we have qubit after the long rate amplification. This is the long amp TAC. And then D, we have the number of nanopore past reads. And so interestingly, okay, in graph A, the qubit of the original sample, the DNA concentration, we're only getting DNA from the actual samples. Most of the samples, including the controls, are well below the limit of detection. And then conventional qPCR, we are seeing amplification of the actual samples 1 to 2 orders of magnitude above the controls. Yay, not squeaky clean controls. There's still like 5 to 10 sequences, 16S sequences in there, but it's okay. So, but after the long rate amplification, only a few of the samples are actually amplified with the long amp TAC. So this is a problem. And so the samples that amplified after long amp TAC were pretty much the same samples that got good nanopore data. So here you can see those samples from the far side of the clean room are really the ones that we got good data from. And we can see that the samples that did amplify above the control samples have much higher proportions of bacterial and some insect reads. In terms of the bacterial reads, just the bacterial reads looks better. So both at the genus and the species level, we are seeing that the true samples are dominated by Acinetobacter and Paracoccus species, whereas the control or kitome is dominated by Cutibacterium, Ralstonia, and Staphylococcus. And we are getting some good species level data that is very distinct from the controls. Now, this was run on the old R9 flow cells, but with the new R10 flow cells from Nanopore, they're starting to hit the accuracy level of Illumina data. And so we could potentially put similar data to this one from those flow cells into a risk assessment model and have it not be garbage. And so again, here you can see from the ordination and in permanovas that those far side samples do cluster separately from the controls, but the ones that really didn't amplify well with the long amp TAC are not. And so there's so many issues to deal with this protocol. Every piece of it's going to need to be QC'd. Because not only does, especially with the PCR step, because not only does it introduce a bias, in this case, the TAC from our qPCR kit is working, but the long amp TAC is not working. And so if any people who are working on ultra low biomass stuff, come and talk to me, let's have a chat about nanopore sequencing. And so we'd like to thank some of our collaborators, especially Scott Tai, who came and helped us with the nanopore sequencing, and Lisa Guan for all her help and work on metagenomic standards for planetary protection. Right. That is
wonderful. Thank you, Anna. So if I can ask all of the speakers in this section to turn on your cameras so we can have a Q&A portion. All right. So the first question here is for Andrea. I have heard of epigenetic age. Do you plan to incorporate epigenetic data into your frailty assessment in future studies?
Thanks for this question. And yes, this is what we plan to do. So we still have to define the data sets. So if you want to collaborate with us, we'd be very, very glad. We're really looking forward for this. So thank you.
Great. And this is to all the AWG experts, all the speakers that have done sequencing. Somebody asked this question, is every sample that is sequenced in space on the ISS, or are samples gathered in space and then sequenced on Earth? And if so, how does gravity and other affecting factors impact the sample?
I guess I could address that. Oh, there's someone coming in our lab. So very relatively few samples have ever been sequenced on the ISS. Like, you know, they have a nanopore sequencer up there, but mostly it's all, you know, sequenced on the ground unless someone has information that I do not. So, I mean, someday in the future, maybe people's experiments, they can get their samples sequenced up there. I will say maybe not the gravity, but just like preserving the sample definitely affects, in order to get it back down to Earth, definitely affects it. And that's no fun.
Thank you so much. Yeah, somebody else have something to add?
Yeah, I just wanted to add to that. We also, like in our study, we treat this also as a potential confounder because you sometimes work with live animal return, and basically like this whole protocol of how the samples are frozen and whether it's live animal return or not, this can potentially confound the results depending on what mission it came from. So it's definitely something that is addressed, but no sequencing done, as Susanna said.
Yeah, for plant proteomics also, we try to freeze the proteomic, the plant in like RNA later. Of course, we had an experiment in our lab where they showed that RNA later definitely affect the protein content and the protein expression pattern, but then is what we have currently and is what we do. So we freeze it about ISS in RNA later, and it is returned frozen. Then we thaw it here on heads and work on them.
All right, thank you all. We have a question here from Matthias. What data do you foresee mined from the literature from your citizen science project?
So for right now, like I said, we were focusing particularly on the cardiovascular outcomes. However, one of the main goals of this project would be to develop a protocol along the way that would allow this to be transferable to any other sort of legacy data set. So this could be other spaceflight data amongst that anyone wants to curate, or additional factors that we've been thinking of as well, is it can apply to other areas, like I said, of legacy, such as rare diseases. You can even use this in natural disasters, again, where the data is low. And so you can go over large data sets for many years in relatively shorter periods of time using the power of this citizen science.
Awesome, thank you so much. I really want to thank all the speakers in this category. Thank you for your time and your fabulous presentations. I know 5 minutes is not easy to cover a lot of information, so thank you. We do have to move on to our next category here, which will be status updates from our AWG leads. And the first speaker in this category is Nate Shevchik from Ohio University, and he will be talking about the animal AWG update. So give me a moment to share.
While she's sharing, I'll just comment on that answer that Matthias gave, which is the other exciting thing about his project is it starts to move us towards evidence-based recommendations for future science. Because I think a lot of times we do things like have expert opinions rather than actually taking a hard look at doing actual systematic reviews of data. All right, thank you, Lavorka. In terms of an update, I have just one slide. So really, this is going to be a high-level overview of kind of what's happened in the last year. I think Lauren did an excellent job kind of explaining what the guiding principles of the AWGs are. So I've broken down kind of the update into some categories, the first of which we'll call guiding gene lab or providing advice or trying to advance the AWG agenda. And so really the main thing that we've done in the past year is work on closer collaborations with ALSDA. So Matthias just gave a nice talk about some of that, and also with the multi-omics AWG. As part of this, we've ended up advocating for merging of gene lab and ALSDA analyses and potentially restructuring the AWGs. And so you've already heard from Lauren that NASA's decided to introduce the OSDR. And so I think that's a very positive thing, at least from the animal AWGs perspective. We also currently have several papers under submission jointly with the multi-omics AWG. So you've heard about a couple of those already today. So Bejin presented, Andrea presented, and Jonas presented on some of those papers. And I'm sure there'll be a bit more about those briefly in the multi-omics update. Otherwise, one of the things that we like to think about the AWGs doing is helping our members. And so in that kind of category, it's really nice to say that 2 of our junior investigators were successful in getting GeneLab analytics grants. So they're both new investigators in their first academic post, so that's great. And we've also had some AWG members get a NASA ground study funded this year. So again, that's great in terms of individual success. And I just want to reiterate those comments that Sylvan made earlier this morning about how delightful it is to actually have had Eliah as our keynote speaker. Obviously, she's come a long way from where she started in terms of starting to work with the AWG. In terms of, I think what most folks have been focused on in terms of the AWGs, though, is what I tend to call our work or our analysis. So before kind of talking about some of that, I would just want to point out that one of the other things that our AWG has done is we've actually tried to spin out other initiatives to support people so that we don't have to do everything just in the AWG. So for example, there was an interest in actually doing single cell work, and we've now kind of tentatively agreed to spin that out into another analysis working group. First meetings have been held, and again, amazingly, that working group has actually got a grant from NASA to do some of this work. So we won't really be monitoring that anymore in terms of the animal AWG. Similarly, the International Space Omics group is continuing to work on protocols and publications. Website for them is there and available. So again, we are going to stop kind of monitoring that as an AWG and let them continue as a great independent organization. Also, we've kind of monitored the ESA Space Omics topical team. That's been a bit more of an initiative across all of the 4 original AWGs, where really this was about trying to get some funding for European researchers to participate in the AWGs, and it's been incredibly successful. Really, the group has elevated omics within ESA. So specifically, they've managed to get omics actually into the science strategy. So that's roadmap 9E, and the link for that is there. The actual recommendations are currently being reviewed at Nature Microgravity. So in theory, they will be fully open access and easily findable without having to have that specific link for ESA. I think for folks in this audience, there won't be any real surprises in terms of the recommendations. It's things like being able to link the omics to physiologic endpoints, being able to look for commonalities across tissues, being able to look for non-commonalities across tissues and species, and of course, actually trying to apply these sorts of techniques in real time. So that kind of gets to the more AI, ML sort of side of things. Space Omics topical teams also had a special issue in Cell Press. There's currently 10 published papers there. Provide the link for you if you want to go through them. Really kind of talks about the history of doing omics in Europe, some individual projects, and some recommendations for future directions. In terms of the actual kind of AWG work within the AWG at the moment, there are several projects in progress. Some of these are with old data, so US-based data in GeneLab. And then we've got some new flight data sets, not all of which are in GeneLab yet, that we're working to start to analyze and get them deposited in GeneLab. And so those will be ESA, UK, and JAXA data sets. And specifically for this year, one of our focuses, I think, is trying to work on increased collaboration with JAXA. And I'll just point out that, you know, they are one of the first space agencies to be putting astronaut data into GeneLab. So they've just in the process of depositing a second batch of that. And that's all I have for this year. Thanks, Lavorka.
Thank you so much. So our next speaker in this category, our next co-lead is Daniela Bezdan. She is from the University of Tübingen, and she is representing the microbes AWG. So go ahead and take it away.
Hello, I'm sorry for the setting. I'm not sure, but we had problems with our internet in our lab, and I'm now in the basement. So I hope you can hear me well.
Sure can.
Okay, the background is a little bit high, but I'm trying to make it short. So it'll be much shorter than I expecting, but it's just like current situations. Okay. So we'll skip a couple of slides because it's very loud here. Here we go. You can see my screen?
Yes.
Perfect. Okay, so again, thank you for this meeting. It was really great so far. I was able to follow it. So in 2003, like 2002, we gained more than, now we are more than 100 plus members at NASA Gene Labs. We're looking forward to double it. We have a lot of more interest, especially with the Inspiration 4 and in the Axiom Station. There will be so many new microbes experiments coming on, so we're really looking forward for new members. And also we have the I4 and the YAXA paper package going. So it has been a very busy year. I think that in the next 2 months, we have much more to report. I received a couple of slides for members of NASA Gene Labs, but also slides which says, please not share it yet. So that's because they're not all accepted. I think as many of you know, because you sent me the slides. So in general, what we're going to do, we're working on ongoing future manuscripts. So of course, the wife Inspiration 4 and the YAXA papers. This is a sentence on brain, too many to count. It's totally true. We are still working on rodent research 6 publication. It's really getting along. Thank you, Nick. Amazing effort here. And we also work in a review the microbial waypoints on our path to Mars. Something which we're really exciting, which is getting really, really big. So there's many stakeholders, and at some point we're thinking of writing a book, another review.
Daniela, are you still with us? Okay. I think we lost Daniela, but perhaps we can move on to the next speaker, and then when Daniela is able to rejoin, then maybe we can finish her presentation. Marco, are we able to remove Daniela's screen share? Okay, perfect. Danielle, are you back? No, okay. So we're going to move on to the multi-omics AWG, and Nathaniel Shevchik from Ohio University will be giving this update as well. So let me just pull up your slides.
I hope I'm back.
All right, Nate, you can take it away.
Thanks, Lavorka, and special thanks to Ashvin for so kindly letting me give his presentation. Just again highlighting the fact that we've kind of moved towards being a bit more cooperative between a couple of the AWGs and trying to find some new ways of working. So there's a picture of Ashvin on this particular slide. And the other thing I'll say is that you can see Ashvin's slides are much nicer than mine, which is the other reason that we like to work with his AWG, because they do all kinds of cool visualizations. Next slide, please. All right, so much like I had a little bit of a thought about the way that I view my AWG, this is an example of how Ashvin tends to think about his AWG. And so I think really the key idea is that we try to integrate multiple data types to advance space biology. But he also has a really kind of defining feature of the group, which he's listed as the main goal here, which is to work as a large community of scientists on exciting and cutting edge space biology projects. And the nice thing about this particular approach is it lets every individual bring their unique skill set to the table. Next slide. And so we've seen a kind of similar sort of graphic to this earlier for the introduction to the AWGs. And so this is an example of what Ashvin's put together for his AWG. I think the main things to kind of highlight here are that it is an international group. And I did see that there was a question earlier about how nationality is tracked. And because this is NASA, we actually track nationality based upon the location of the organization that you happen to be located at, because that's how the export control rules work. And on that note, if you look at the kind of logos of some of the organizations that you'll see involved here, there's a nice mix of academic groups, national organizations, and industry. Next slide, please. In terms of past work, I think there's kind of some general themes that we can see. Up in the left corner there is the Cell paper that really focused on mitochondria as being a key hub of the response to space flight. We can see below that and to the right of it that there's kind of a look at actually trying to see what the impact of radiation, both alone and in combination with other factors, is on biological systems. And again, in terms of the usual suspects of things that people have worked on in the past, in the bottom left, we can see that the group's been looking at changes with muscle. But I think the really exciting thing about the group is that it's working in looking at new directions and new problems. So for example, earlier today, we heard from Bejim about things to do with endocrine and female health. And we also heard from Jonas about using machine learning approaches. Next slide, please. To go into recent papers and things that are currently under review, I think it'll be really no surprise that there's a fair number of things that are using Inspiration-4 data. So for example, we heard Bejim present her paper that's under consideration at the moment on estrogen and insulin signaling pathway alterations across species and across tissues. Next slide, please. We also heard from Andrea about his exciting new concept of, we know that there are similarities between aging and spaceflight in terms of biological changes. And an emerging theme or idea within the aging community is, can we actually assess frailty and use that as a marker? So he's started this very exciting initiative to see whether or not there are markers of frailty associated with spaceflight and whether that relationship between aging and spaceflight is conserved into this sort of concept. Next slide, please. We also heard earlier from Jonas, who talked a bit about a portion of this paper that's actually looking at skin changes. And we've also heard from some other folks about looking at skin today. But I think for this particular paper, what I'd really like to highlight is what I find super exciting about this project, as shown in red. This actually has a fair number of Gene Lab for high school participants who have actually participated in co-authoring and co-creating this particular paper. Next slide, please. We also heard earlier today from Andreas about trying to look at similarities between sarcopenia and spaceflight and trying to think about, can we apply things from the clinic on Earth to trying to solve problems in space. Next slide, please. And then, of course, at the beginning, we had our keynote speaker, Eliah, talking about the fact that we've entered this commercial space flight and commercial space flight participant age. And so one of the things that the AWG has been doing, which is similar to what Trish has been doing and what's similar to some of our international partners are doing, is starting to think about the ethics and also the operations of how do you actually think about doing experiments with private astronauts effectively. Next slide, please. We heard earlier about the idea of there being an epigenetic age. And so again, it shouldn't come as any real surprise that the group is starting to look at epigenetic changes or structural changes to the DNA with radiation. And so this is a particularly exciting area because I think a lot of times people don't realize that we can actually look at the structural DNA data and actually look at chromosomal regions to actually think about why mechanistically we're getting these changes in transcripts that we are. Next slide, please. Again, continuing on the theme with focusing on female health. I've also got another article that's under consideration. Next slide, please. And again, in terms of other types of data sets, group's been looking at microRNAs and how DNA damage or other types of damage induced by radiation may be modulated or may modulate microRNA expression. Next slide. And lastly, in terms of papers that we just wanted to highlight from the group's also worked into starting to look at cardiac health, which again is a fairly exciting thing because we know there are changes in the cardiovascular system. We've heard a little bit about that, and we know that there are changes to muscle. So it's nice to start to be able to expand this to cardiac muscle. Next slide, which is the last slide. There are many future projects. Just thrown 3 of them up on this particular slide. One is kind of keeping with the theme of studying mitochondria and looking at what goes on with mitochondria across different tissues within one organism and across species. Also continuing with the frailty project that Andreas presented and continuing with the endocrine and the female health theme. But I think the really exciting thing here is that this group is ready to work on new things. They're a great group. They're a really collaborative group. And so really, if you're looking and joining one of the AWGs and you've got an idea, but you don't necessarily know how to make it happen, this is a great group to come join and really kind of get you on your way. You can see from all the papers that are currently out, very productive, very supportive group. And thank you.
Thank you so much, Nate. Appreciate that. So it sounds like we have Daniela back on. So Daniela, if you are able to hear me and able to share, why don't you go ahead and give it a try again? Marco, can you give Daniela permission to share, please?
She's a co-host, so she should have permission.
Now it's working. Okay, I will share the screen, but I'm not switching on the camera. I'm so sorry. The maintenance is here in the cellar. We try to fix it, but looks like the whole institute is out of Wi-Fi. Sorry, it's super unexpected, not planned. Okay, I try again. Bear with me. Trying. Okay. So I will skip a couple of slides. So ongoing future manuscripts, we did some brainstorming and we have exciting projects. So the moment the Inspiration 4 and the JAXA papers are out, we will want to concentrate more on mark contamination. We want to work on DNA alignment scales, monogenomes, needs a beta tester. Viral phylogenetic pipelines need beta testers. So if one of these topics are speaking to you, please join Microbes Lab, and we want to explore in that direction. Also, there's a couple of things which Braden asked me to share. They're co-infected microbiologist for working with carbon capture applications. This is something we're going to investigate as a group as well. And the other one at camp is new metagenomic. It was a metasub. GitHub camp got an update for the assemblies. This is all we're going to investigate it. And I think, sorry. Okay, it's hanging. I think, I'm not sure if you can see the slides anymore.
We can see slide RR6 fecal taxonomic whole genome sequencing.
No, it's hanging. I'm already in the next slide. Okay, Lavorka, let's skip it here. These are like now, I think these are experiments from Nick and from Gilmore, but that's about this. Let me share the slides and by email, and we fix the problem, and let's use the one time, because I don't think it's not moving. It's not moving on my side. I'm so sorry.
Right. Thank you for trying. I appreciate it. All right. So our next speaker will be Christina Johnson. She is from NASA Ames Research Center, and she'll be speaking on behalf of the plant AWG. So Christina, if you are on, can I please?
I am here. I hope it's working. Can you hear me?
Yes, it is. Absolutely.
Perfect. Let's share and hopefully everything will go well. Oh, of course, I need to press the share screen button first. Imagine that. Sorry about that. Okay, is this showing my slides?
Sure is.
Perfect. Okay, so hi, I'm Christina, and I'm part of the plant AWG, and I'm going to be sharing with you the progress of plant-based biology omics to date. This was put together by me, Kristen Peach, Colin Cruz, and Richard Barker. So here's a picture of some of our plant AWG members. There are 84 of us, so that not everyone could fit on the slide, but it's a really great international team doing great work. So we have about 2 meetings per month. Sometimes we have fewer because of various reasons, holidays or symposia or whatever. But one of these meetings has group updates, a focus on announcements, and some big overarching things. That all happens on Teams. And then we have a Zoom meeting at a different time once a month, where we have breakout sessions with focus groups on specific projects. So over this past year, from March through March, we've had 20 meetings. And we had, not sure if you can see my cursor or not, but we had 12 of these meetings had 10 to 13 attendees. 6 of these meetings had 14 to 19 attendees, and 2 of these meetings only had 6 to 9 attendees. So that's actually really good attendance for the plant biology world. Not sure about the other AWGs, but we're really happy with those numbers. In general, we tend to have about 12 participants per meeting. The smallest was 6 and the largest was 19. So we noticed that there are higher attendance during academic breaks, and so that's something to keep in mind in the future. An average AWG member attends 3 meetings over the course of the year, and that's because a lot of our work is done asynchronously. So just sharing a few recent GeneLab enabled plant publications. This was published by Ariel Hughes and John Kiss. This covers really great omics studies of plant biology and spaceflight. So it had a bunch of these recent studies that they just put together and reviewed, and really showed off the hardware with great graphics, great publication. And then also Alexander Myers and Sarah Wyatt put together this plant-based biology in the genomics age, which is a really great review plus future insights about what we'll need to do in the future. So really great papers, highly recommend you read them. But then there's this one that we've been working on for 2 years now, I think it is, a meta-analysis of the spaceflight and microgravity response of the Arabidopsis plant transcriptome. And this is the one that we've been working on as the AWG together spending a lot of time. It has a lot of authors, and it's in proofing stages. And we were really hoping to be able to share a link today, but not quite yet. Hopefully in the next week or two. I don't want to jinx it. We also worked on the white papers and submitted a bunch of white papers. We worked together on this research campaign, Space Biology Reference Experiment Campaigns for High Fidelity Plant Physiology. And then we also had a lot of contributions by AWG members. So there were 3 contributions. 3 of these papers were contributed by AWG members that were related to plants in space. And then there were 2 white papers submitted on topics related to the AWG mission that Plant AWG members participated with. And this was Omics and Open Science, a platform and approach for the future for space biology, and also one about including disabled population with inclusion efforts. So next steps in the support of space exploration. So we already have 62 plant data sets from 6 different species in the OpenScience.archives. That's a lot compared with the past. We're going to be growing so much more. These are just imaging lab repository, but we are now looking at how the OSDR is accepting non-omics plant data. This is huge. This is going to change things. We're going to be able to start looking at plant phenotypes. We're going to start looking at the morphology and development of plants in relation to that omics information that we've already been collecting. So this is a very exciting shift, and I'm excited to be a part of it. Keeping in mind the decadal survey is going to be coming out this summer. And so we're all looking forward to seeing what they took from the white papers we wrote and what are going to be prioritized for the next 10 years. And we're going to be working with that. So also I went back and I looked at Plant AWG membership overall and activity in the last year. And we have 86 participants in the Plant AWG. 47 didn't attend any virtual meetings this year, but many of those contributed to papers. Some of them viewed our recordings of the meetings, and every one of them that I contacted are all enthusiastically available to the Plant AWG as subject matter experts. And they're just waiting for us to reach out to them and ask them questions and welcome their participation. So while they may not have the time to attend the meetings, they're contributing to publications, they're making an impact in our group. And I see that as untapped potential. And it's really exciting. 38 of our members did attend at least one meeting. So this is, I don't know if you can see my cursor, but the larger chunk here that's in gray was the 47 that attended no virtual meetings. And then this little green part is the 38 that attended at least one meeting. And this is a breakdown of those 38. So we had 5 people who went to more than 15 meetings. I am so impressed with the people who could go to that many meetings and made time for that. They have been the backbone of our group. So 13 of these folks went to 5 to 14 meetings, and then 20 went to 1 to 4 meetings. And even having them at one meeting makes a huge difference, especially if they attend one of those meetings where having the breakout sessions. It's really great how they're participating and being a part of things. So I think moving forward, we're really going to be working on engaging with the Plant AWG in a way that they need to be engaged with. Inviting labs to give scheduled periodic updates during AWG meetings will be a way to get kind of a, hey, what's going on with the Wyatt lab? Oh, hey, what's going on with the Mars lab? What's going on with the KISS lab? Let's hear all of these great things. Welcome teams we're going to start seeing teams that are sharing non-omics data, and we need to welcome them with our open arms, whether that's at the Plant AWG or a separate ALSDA AWG that's focused on plants. Somehow we're going to have to welcome them and incorporate them into the discussion, because there is going to be a lot of crossover with our work. We'll want to advertise breakout sessions in advance. A lot of times we had breakout sessions this past year where it was like, oh, we don't quite have the people we expected. Oh, we forgot to reach out to them. We better do that better this time. And also something that came up was having a writing club meeting twice a year where we could get together with our current manuscripts, look at them as a group, seeing what contributions people have to bring to those. Just getting another set of eyes makes such a big difference. Another thing we noticed as we were looking at our email list, some of these emails aren't current anymore. We've lost track of certain people. So we need to go find them and make sure that they want to be a part of the AWG still, and make sure that their emails are current. And also something I'd really like to do is ask present and past members how they want to improve the AWG, how they want to personally engage with the AWG. And I think that will make a big difference to improving our productivity. And that's it. Thank you very much.
Wonderful. Thank you so much, Christina. And our next speaker in this category is Ryan Scott, and he's from NASA Ames Research Center. And he is the AWG lead for the Ames Life Sciences Data Archive AWG. Go ahead, Ryan.
Great, thanks. I believe the screen is sharing, correct?
Yes.
Yes.
Oh, wonderful, cool, thank you. Gosh, Christina, I really liked how you did a breakdown of the AWG membership and meetings and attendance, and how to go back to previous AWG members. And it speaks very closely to what the ALSDA AWG has been going through. So the ALSDA AWG started in the summer of 21, if I remember correctly. And it's been an interesting journey. So I'll go over that, and just to start off with a snapshot of the various members, just a portion of the kudos of AWG members. But yeah, one thing we are finding is, depending on people's expertise and their area of interest and subjects, it's an ebb and flow. I usually figure there's generally, when we have an AWG meeting, we haven't had one for the last couple of months for the ALSDA, but it's because we've been having several meetings a month, subgroups, and we decided not to we should talk about this more. I just really enjoyed your thoughts, Christina, on what to consider. So in terms of just the ALSDA overview and sort of the big picture, when you think of GeneLab, you think of the molecular omics data, transcriptomics, GWAS, proteomics, metabolomics, transcriptomics, that type of thing. But what about the rest of the data? And so this is one of the things that we think of, which is one of the reasons ALSDA and GeneLab had the integration starting, where I guess it would be summer of 21, right? So that animal data, this could be rodent, it could be fruit fly, C. elegans, a number of different assays. So it could be behavioral assays, it could be tonometry, it could be Western blot, it could be spectrophotometry, it could be MRI, it could be ultrasound, of course, microscopy, plant data, all the way from the images to, that's why I was so keenly listening to Stephen's talk on this hyperspectral imaging, but also photos, or maybe you have the soil and growth nutrient profiles. Of course, human data, as human data comes into OSDR, we are having standards for those data, of course, as well. And this could be, starting with the Inspiration 4 for non-omics data, we are looking at cytokine panels, comprehensive metabolic panels, and complete blood cell counts, but also having the type of data on the human data set, so that those also can be made reproducible and findable and have a good, that type of thing. But also thinking about microbial data, yeast, algae, especially with the recent BioSentinel mission that was deployed on I almost said Apollo, Artemis. And so when I think of all these types of data being part of ALSDA, you can think of these as any of the tabular, the text, the imaging, the video, or the code, always striving to not just have the tabular results that you probably put in a pub, but also the raw data. That's going to be the sort of the striving gold standard that we want to go for, so that, if there's questions on the histology, or questions on the Western blot, or questions on the imagery, you can always go back and look at the actual raw bioimaging, or whatever level of data it came from. And in terms of, and beyond the scientific data, also the telemetry. So I know at the beginning of today's event, Lauren did mention the environmental data application, but which right now is largely focused on helping, on shining a light to the telemetry related to rodent research, but we do have plans to, I know there are plans to extend that. So, just a recap on what is OSDR. It's really the combination, including these three different groupings forming the OSDR, which now has the single submission portal. We have the user interface. Data are maximally open access, so there are necessary controls for sensitive data, and data are maximally fair. And so what is the ALSD AWG been largely focused on? Because this is a new chapter of data types that we're curating, making available to the public, the AWG has played an unbelievable critical role helping to standardize the ingress of data, as well as helping us standardize the egress of data outside of OSDR. And so, for example, in the submission portal right now, these are initial versions. If you've gone in there and tried to play around with these, they are initial versions of different types of assay metadata. So these are aspects and parameters about the device, about the reagents you used in the assay, the settings of a microscope, or maybe aspects of a behavioral, it could be like a behavioral rodent test, right? And so this is what largely the AWG has been helping provide feedback on, are these scientific standards, so that when the data gets released, you can really have confidence in the science acumen of the data. So a really, really great spin-off activity that Matthias just presented on a little while ago is that Solstice Citizen Science Project, which presented a poster at HRP IWS, as well as 2 posters at the recent Canadian Space Health Research Symposium, as well as 3 grants have been submitted to NASA and the Canadian Space Agency to make that citizen science project more official. And of note, here are several current data sets that are now live on OSDR that came through this pipeline of curation and data collection via the which was connected to the ALSD AWG. So one other thing that ALSD AWG has been providing feedback on is in the coming year, there are plans in OSDR to expand the architecture to really focusing on bioimaging and video data to make these maximally open access and reusable. So, we have the investigation sample and assay, and sample metadata pretty soundly covered within the repository. But one of the things that needs to be expanded are the assay and modality metadata, so that they can then also have file and format conversions when these types of, it could be microscopes or photography, or it could be MRI, or it could be CT scans, having file format conversions. It does seem that bioformats, which is actually just an ImageJ plugin that we've developed a relationship with the open microscopy environment that developed the bioformats tool. So looking at that, and then also thinking about the data itself on the imaging side, so that raw, but also you could have annotated data, or you could have segmented data. And so different levels of process data, how to handle those to make them also findable and uniquely available through the API, but also for the public and for your own reuse, because there's this amazing renaissance that's occurring in the bioimaging world. So we also don't want to have people download a terabyte of image data if they want to go look at a microscopy data set. So figuring out a different data visualization tools, and also ways to chunk the data rather than have to do the direct downloading and for visualization. And so this is one of the other activities the AWG has been involved in, spanning from Jackson Labs to Quarup Limi, Bioimaging North America. European Bioinformatics has their own image archive called the Bioimage Archive, which is great, and in a pretty similar boat to where we hope to be within the next coming year. So which only focuses on images. So that's another aspect of what's coming up in the future. Was this past year? Yeah, just last year, right? We had the first ALSDA AWG publication, which is excellent. The Human Systems Risk Board, centered around NASA Johnson Space Center, recently released a series of what they call directed acyclical graphs, which are trying to take evidence-based data and graphs. So nodes and edges were produced into these 36 or 37 unique directed acyclic graphs. And they sort of, now that they had all these SMEs produce these graphs, and so what they wanted was to actually begin to validate them with actual data. And so several different bone data sets that were in OSDR that are currently either available or being released. 2 of them are available, 2 more are about to be available, were shared and then put out for this publication as a proof of concept on ways to validate the DAGs, which can be used for eventual risk quantification for astronaut health and individualized risk quantification. If anyone has those links, it'd be great for someone to put those. I see Rob's here, so it'd be great if someone could share those links in the chat. Another AWG collaboration was what Val Fajardo pointed to was, and also which ties to ABZU, which I know Jonas was mentioning, is the explainable AI activity on muscle loss. After Val submitted the RR9 calcium uptake spectrophotometry data, there were already RR9 data and RR1 data in, of course, from GeneLab. And so it was a really nice exercise to be able to integrate those data and then have new results that have real biological implications. So those are the data sets connected to there. And I don't know if Henry Cope is still around, but another thing I'd like to think is an AWG activity is having developing a pipeline with the computer vision model tools and transformer tools, because now that we have the omics data from GeneLab and the non-omics physiological data and imaging data, we have an ability to use some of these more machine learning tool models in an image processing pipeline for then someone from the public could go select these imaging tools and then look at the data, select an imaging tool, and then merge the data for even higher level analytics. So it's a pleasure to be able to collaborate with Henry recently on that. I'm not going to go over these in great detail, but just to give you a sense of what goes into these assay metadata configurations and the entire sort of overview and workflow. So this is to make all these various assay configs, we call them for short, that we do have a process established, and it just took a phenomenal amount of effort and feedback and revisions and sub meetings and breakout sessions, and but we also, for the results itself, to have confidence in the tabular results, because right now every data set that comes from us from a PI, the tabular results, every single data set is individually unique. These data don't come to us standardized like raw sequence counts or even process data. A lot of these are just data sets that are in Excel or CSV format that were used for a publication. So we've come up with these data templates and standards for the results data, which we're working on. And then lastly, we do have the ALSDA curation and public release pipeline that leads to before we actually release a data set. So it's one of the things that the AWG has really played a key role in providing feedback for. So I won't go into too much detail, but I thought it would be worth mentioning, walking through one config. This is for the elevated plus maze assay. This is a rodent assay. And so just to give you a sense of, after a series of meetings and asynchronous edits on a shared Google Doc, we then had a few different breakout sessions within the AWG monthly meeting for ALSDA. And then that December 2021 is when there was a final consensus, like, this is a good EPM, elevated plus maze assay. And this involved ontology selection and sort of always trying to have gold standard literature drive the decision making. But then having to think about the arrangement of the assay, especially behavioral, compared to other different assays, the arena, the objects, the various different characteristics or structure sizes and specs, to other different characteristics about how the assay was performed. I will encourage you to just remember that arena and objects for a moment, like the shape of the arena or the size of the arms, because I'm going to show it to you again in a second. Of course, then you have the testing environment, and this was big scientific implications if you don't have the metadata, for example, on even the cleaning solution of the platform that you used for that animal. That can throw off the results of that behavioral assay because of the role of olfactory being so dominant for rodents. So and then here's an example of that standardized template. And then just like I said, to remember that example of for the arena and objects. This is an example of about to be released data set, which is using that elevated plus maze assay. And like here's the overview, the various factors. We have the various protocols that were part of this study. Here's the sample table, which a lot of people probably recognize if you've perused OSDR before. And here's an example of that EPM assay config in practice. Here's the arena description, and you can actually see what's the texture and materials, you know, what's the size of the arms, the dimensions, what was the shape of the arena, right? So you can sort of see why we had to ask the SMEs, what do you need to have to make this data the minimal, absolutely necessary information. And now you can sort of see what it looks like in practice. So, and then actually the data itself, thinking about the template, this is an example of the types of tabular data that we get. And then what we do is we transform it so that there's one single tab, it's out of Excel, it's in a CSV, so it's easily exportable. And it might sound simple, and this is really important. All of the samples are in a row, whereas all of the parameters that you've measured are in a column with maximum, and we have an AWG standardized nomenclature on top of it. And this is really important for future PIs that submit their data, to always strive to have your data in this type of format for all samples in a row and all columns in the parameter as a parameter. So there might have been a little, I just probably should have mentioned this earlier in the day, but people might be wondering about the numbering structure that's currently going on in OSDR, because we have our new OSDR numbers. So this is just a snapshot of what all that means. So yeah, once again, thanks to all the AWG members, and appreciate it. Appreciate your time. Thank you.
Awesome. Thank you so much, Ryan. That was a great overview of the ALSDA. And we have our final speaker of the event. We are coming full circle back with Lauren Sanders, who is at NASA Ames Research Center, and she will be talking about the Artificial Intelligence Machine Learning AWG. Take it away, Lauren. Lauren, are you speaking? We cannot hear you. Still cannot hear you. Okay, I think we're going to give her a moment to come back. I think she left, and will rejoin momentarily. If you have any questions for Ryan, feel free to put them in the chat or on Conference IO. There we go. We can hear you. Welcome back.
Thank you. I don't know what happened. You could hear me this morning. All right. Can you see my slides?
Yes.
Great. All right. Thank you, everyone who has stayed with us to the end of the day today. I'm going to be providing the update for the newest AWG, the AI/ML AWG, on behalf of myself and my co-lead, Dr. Jaden Hastings. So when we started the AI/ML AWG, we held a working session to develop 4 main focus areas for our group for the next 1 to 3 years. These are aspects of AI/ML and space biology research that the community and the scientists in our group felt were the most important for short-term progress. So the first focus area was we want to work on enhancing the AI readiness of the data and metadata in the Open Science Data Repository, or OSDR. This work is going to focus on generating guidelines for standardized AI-ready dataset structures for the data system. The second focus area is we would like to work on developing algorithms specifically that will work with the constraints and challenges that we face in space biology research. So this area is going to focus on gaps in current capabilities and developing benchmarking tasks for new algorithms that are developed. The third focus area has an emphasis on self-driving labs, which are these automated closed-loop AI-enabled experimental platforms. So we're here going to prioritize physiological, biological questions for automation and work on integrating multiple automated experimental platforms and getting comprehensive experimental readout. And our goals are to help develop the capacity for in situ analysis, so on a spacecraft or on a CubeSat, and active learning, so an AI system that can react to input. And the fourth focus area is for supporting trust, ethics, and explainability for AI in space biology. So it's particularly important to us to help train the new generation of AI investigators to conduct ethical experiments and to help establish consensus and expectations for ethical research standards for the use of AI/ML in space biology. So the membership of our AWG has a variety of priorities and goals, including 65% interested in gaining skills and knowledge sharing for research, almost 25% wanting to learn more about the data that's available through OSDR, and then additional groups who are interested in networking for career development and publishing scientific papers. We currently have 2 active subgroups in our AWG. So one of these is on self-driving lab technology, led by Dr. Adrian Horfrost, and one on research and writing, which is led by Dr. Jaden Hastings. The subgroup on self-driving labs will have its first meeting next week, so I will just review the goals of this group at today's update. We envision a cycle in which we train a model on labeled training data. We then use an acquisition function to design sample selection and an experiment from an unbiased design space, automated human-in-the-loop testing of the target outcome, and then adding new labeled examples to that original training data, and the cycle then continues. The motivation for this work is to increase sample number through enabling higher throughput experimentation in space, increase repeatability and reproducibility via automated processes, and free up human time. Now to switch gears and give an update on the other subgroup on research and writing. The motivations for joining this group are diverse, from wanting to engage in brainstorming and discussion to being interested in collaboration and writing papers and proposals. The core philosophy of this subgroup is to enhance capacity and collaboration, provide a dedicated time for people to get together and advance specific projects, identify existing knowledge and gaps, and support all of our group members through professional development. The current topics being explored and written about in this subgroup are compute power in orbit and beyond, next generation biocomputing, space medicine and automation, and ethical challenges for AI in space biology. And as a group, we're currently working on at least 2 review papers, one of which is focused on ethics and one of which will focus on computing in space. So as a group, we're very excited about the opportunities ahead of us, and we would love to invite your participation. Please feel free to reach out to me via email or sign up for our AWG using the sign up form in the chat. Thank you.
Thank you so much, Lauren. And this was our last presentation and concludes the 2023 Analysis Working Group Symposium. Again, I want to give a big shout out and a big thank you to all of our speakers for putting in the time to put together these presentations and share their work. I want to thank Blue Marble Space for their sponsorship and providing this platform, as well as Marco for helping us deal with all the technical challenges during this event, and also all of the participants. We thank you for joining us. We thank you for listening. And again, if you are interested in joining, fill out that sign up form. The AWGs would love to have you. So with that, thank you all, and until next time. Goodbye, everyone.